but is not. Thank you, Katie. I appreciate that. Excellent. So
communication at the muscular junction.
Yep, hold on. I wanted to come back real fast. I want to, since I wasn't recording, make sure we have the schedule thing on there. So there you go. Thank you for catching that. Excellent. All right, so there's a schedule. So anybody's watching this can see that. And then excellent. All right, let's just go ahead and delete this too. All right, perfect. So say that again to me, Laura.
Communication at the neuromuscular junction.
Excellent. So we are talking about muscle contraction. And as we talked about, part one is communication at the neuromuscular junction. Excellent. Again, when thinking about this, it helps us to organize our thought process as we're doing this. So what was the goal of part one again?
To convert the neural signal to the muscle.
Excellent. So, or more specifically, neural action potential into a muscle action potential. And let's add one more thing to it as well. What was the first step in the process of communication at the neuromuscular junction? Excellent. Haley's got it. Make the decision. So the very first step is that we make a decision. And what was the last step?
Make a decision to stop.
True, if we were relaxing it, yes. But in this case, I'm thinking more in terms of the initiation process. If we are trying to get a muscle to contract, if I asked you the essay question, describe initiation of communication at the neuromuscular junction, you would start from making the decision. And where would your essay question end?
A muscle action potential was made.
Excellent. All right. Alina, you guys are all have right there. Absolutely. We depolarize to threshold. We produce the muscle action potential at the motor end plate. And does it stay there at that motor end plate? No. And it spreads down the sarcolemma. Excellent. So that, again, isn't obviously the entire process, but it is a good reminder because remember, this is a continuous process. So when we get to part two, which is where we're going to start today, and we'll talk about that, and we'll talk about the goal of step two. But even though we know nothing else about step two, guess what the first step of step two is, of part two is?
Initiation.
Traveling on the sarcolemma.
There you go. Exactly. Remember, as we talked about, the end of one part is going to be the beginning of the next. So the very first step of our second part of this muscle contraction is going to be to produce the muscle action potential at the motor end plate. And it spreads down the sarcolemma. Because this is a continuous process, what is the last step of part one is going to be the first step of part two. Now, of course, let's add some more vocabulary. What is part two called?
Excitation, contraction, coupling.
Excellent. And what did we say the goal of that one was?
The goal was to move the regulatory proteins by using calcium.
I like that. Use calcium to move the regulatory proteins. Excellent. All right, so that's our game plan for this first part of the class. We want to use calcium to move the regulatory proteins. We want to now do excitation, contraction, coupling, and we know it is going to start from where part 1 ended, with the production of that muscle action potential and that muscle action potential spreading down the sarcolemma. All right, questions on this? All right, excellent. Just like we did for the previous one, I want to go over this twice. We'll do it with the pretty pictures from the textbook and the slides, but I also want to do it on the board as well. So let's go ahead and use the whiteboard to discuss this process. And again, our goal now is to talk excitation, contraction, oops, coupling. And again, this is part 2. And even though we literally just said it, it's always good to be thorough. So what is our goal for this step again? Should be easier since we literally just did it like a minute and a half ago.
Use calcium.
Excellent. We want to use calcium to do what?
To move regulatory protein.
There you go. Excellent. All right. Now, like we did yesterday, we want to start first with the anatomy. All right. So obviously this is taking place inside of our muscle cell. So let's draw some of the anatomy of our muscle cell here. No, don't want red. There we go. Where do I want to do this? Let's do this here. Oh, that's not the color I wanted either. That's what I want. Okay, excellent. Now, let's not do that. Okay, excellent. So here is our motor end plate with its invaginations. And of course, associated with that is our synaptic end bulb that does not touch it, but we know has vesicles inside of it. This has voltage-gated calcium channels. This has our chemically gated cation channels. Right, everything we talked about for step 1 all occurred at this tiny little structure that is here located associated with a small portion of our muscle cell on the sarcolemma. I'm going to need to make this smaller, aren't I? Yes. Excellent. So we have our sarcolemma here. Now we know something about the sarcolemma. Actually, we know 2 things about our sarcolemma. We know our sarcolemma is specialized to produce and propagate an action potential, make and spread an action potential. But we know something else about our sarcolemma. What else do we know about our sarcolemma? Why do I have a gap here on the surface of my muscle cell? Because what happens to the sarcolemma?
The triad forms.
True, although the triad has one component. There you go. Of course, we're not going to want to use the term T-tubule when we're writing it out on a lab exam, for instance, right? Because no abbreviations. But you guys are getting there, absolutely. We have these big folds, these big invaginations of the sarcolemma called the transverse tubules. Absolutely, excellent. So we'll put that transverse tubule there. Of course, you are absolutely correct. If we were writing this out in an essay question, once we've written out transverse tubule once, or if you wrote yourself a little key at the bottom, then you could abbreviate it T-tubule. Excellent. And as Laura pointed out, this T-tubule is one of 3 things that make up a triad. What is the other thing that helps to make up that triad?
The cistern of the reticulum, something.
Excellent. Sarcoplasmic reticulum. There you go. Exactly. Which, remember, as we talked about, is that specialized smooth endoplasmic reticulum. Now, we know it drapes over the myofibril, but I'm just going to draw it as a little bit of a straight line here. But as we also pointed out, there is a large enlargement to it right next to the transverse tubules that we call the terminal cisternae. So let's go ahead and put some more labels on here. This is the sarcoplasmic reticulum. Excellent. This is, I think I have a little more room on this side, so I'll put it here, a terminal cisterna. Excellent. And these three things collectively are known as the triad. So I will put that, I'm going to put that. I'll write it, I can write it right here. And then with a very thin highlighter, we will remind ourselves that we are talking about, let's do it this way. Like that. And then I can cheat and bring my triad up here. Perfect. Excellent. All right. Now, again, I've drawn these as a simple thing, but we know it is sitting on top of a myofibril. So we know over here we are going to have that special contractile organelle known as the myofibril. However, what did we say the organizing unit, the structural and functional unit of the myofibril is? The myofibril is made up of a repetitive pattern of precisely aligned proteins. And what is that precisely aligned protein structure called again? Excellent. It is called a sarcomere. So let's draw a sarcomere to remind us what something like that looks like. Put that out of my way. What forms the boundaries of the sarcomere?
The Z line?
Right, a Z line or the Z disc is the protein. The shaded region of the organ that it forms is the Z line. It's not a bad thing to use the terms interchangeably, but the protein itself is indeed the Z disc. So let's go ahead and draw a Z disc here. And another Z disc here. Excellent. If I remember correctly, this sarcomere has two contractile proteins. What are they again?
Actin and myosin.
Actin and myosin. Now, one of them is a big, thick filament. What was that big, thick filament called again? Which one was the big, thick filament?
Myosin.
Myosin, excellent. So we have this big, huge, thick filament. I'm exaggerating the size of this because it will meet our needs. We have that big, huge, thick filament at the center. And as we know, it has all of these myosin heads coming off. Now, I'm not going to bother drawing the myosin heads. I'm just going to draw the necks of them. But we know those myosin heads are there. And those myosin heads want to grab and to pull on the actin. So 3 this way, 3 that way. That works. Although I don't like the placement of this one, so let's move that there. And that one there. Perfect. Excellent. That one's too long. All right. Excellent. And we know that they are attached, not attached, but they want to attach to our actin. And again, I'm going to exaggerate the space in between them because it meets my needs. Excellent. We know there are 2 more regulatory proteins, two more structural proteins, the titin, which is going to come and help to stabilize this in place, the myomecin, which is in the center, but they don't really play a role in this, so I'm not going to bother drawing them because I don't want to cloud up our image too much. And let's label all of this stuff. This down here is our actin. This over here is our Z disc. This over here is our myosin. Excellent. Oh, hey, let's be clever. Put that there, make that white. Does that work? Yay, hey, look at that, I'm clever. All right, perfect, excellent. Now, we know that there are also regulatory proteins. What are the two regulatory proteins called?
Troponin and mytroponin.
Yeah, troponin and tropomyosin.
Tropomyosin.
Exactly. So if you remember, the tropomyosin is a long thread-like protein that basically blocks the myosin head from binding to the actin. So let's cheat and grab this and move that up here to remind us that that is the actin. And then this is our tropomyosin. Excellent. Now I need that to be black again. Excellent. But when we want to move it out of the way, we know that there is a globular protein on it, and I will just draw one of these on each of them. There are more than that, far more than that, but again, one serves our purpose. And these glob-like handle proteins are the troponin. All right. So troponin right there like that. Tropomyosin there. Excellent. Actin, Z disc, myosin. I can just move this up here. All right. Questions on any of that? All right, excellent. So now, oh, it's wrong button. There you go. Do you see the picture? Okay, perfect. So whoever's mowing their lawn, please stop, or at least mute your not your camera, your microphone. All right, excellent. So here is our structure. Here is the anatomy. This is where we started the last time as well. So now that we have this, we can talk about the process, and we'll do it again in blue, just like we did for the last one, of initiation of excitation contraction process, coupling. So for the process of initiation, and let's make this a teeny bit bigger. Excellent. Of course, as we know, it is going to start with the muscle action potential. Excitation, contraction, coupling begins where communication at the neuromuscular junction ends. So remember, we have over here at our motor end plate, we have produced that big, huge, oops, nope, blue. That big, huge positive signal, that muscle action potential. And we have spread it down the sarcolemma. I guess I'm gonna have to make that smaller. And it spreads down the sarcolemma. So we produce that muscle action potential and it spreads down the sarcolemma. Now, not only does it spread down the sarcolemma, but it spreads down the transverse tubule as well. So our muscle action potential spreads down the T-tubule. And since I've written it out once, I can go ahead and abbreviate it here. It's going to spread down the T-tubule. Now remember, the muscle action potential, like the neural action potential, is a big positive signal. It's a big, huge wave of water. And if you're sitting next to a big, huge wave of water, what's going to happen? You're going to get wet, absolutely. So when we have that big, huge positive signal come down the transverse tubules, not only is that going to make our transverse tubule big and positive, but it's going to splash over onto the terminal cisternae as well. So our entire triad is going to get big and positive. And is there a term for getting big and positive?
Polarize. Depolarize.
Polarize, absolutely. So what this does is this depolarizes our triad. Which makes me realize I forgot one of the most important anatomical things we need to talk about. So we're going to depolarize our triad. We can put this here. Why do we care that our triad is depolarized? Because there are two more things that we forgot to talk about. What is the function of our sarcoplasmic reticulum?
Store calcium.
Excellent. Function is to store calcium. Remember, obviously, the place in your body where you store the most calcium is the matrix of our bones. But the second highest concentration of calcium in the body is located here inside the sarcoplasmic reticulum. For instance, one of the places where there is the largest collection of criminals outside of Washington, D.C. is Folsom. Right? Does that mean that you shouldn't go to Folsom at night? Maybe don't open a store in Folsom? There are a whole buttload of criminals in Folsom, but where are most of them located? Inside the prison, absolutely. And if they're locked away, they can't cause any problems. And it's the same thing here. We know calcium makes cells do wonky things, but if it's locked up in the sarcoplasmic reticulum, then it can't do wonky things. It can only do wonky things if it gets out into the cytoplasm. In fact, one of the things that our sarcoplasmic reticulum has to try to, oops, don't want that, to try to make sure that our sarcoplasmic reticulum doesn't do, that our calcium doesn't do wonky things, is that it has a large number of calcium pumps. These calcium pumps take calcium and move it into the sarcoplasmic reticulum. So we have these calcium pumps that are going to take any calcium that is in the cytoplasm and lock it up inside of our sarcoplasmic reticulum. However, if we want this muscle cell to contract, eventually we're going to need to let the calcium out. And the way we let the calcium out is by having a bunch of special channels on our sarcoplasmic reticulum and on our terminal cisternae. And these channels are ones we're familiar with. Voltage gated calcium channels. So we have these voltage gated calcium channels all over the surface of the sarcoplasmic reticulum, all over the surface of the terminal cisternae. So we have those calcium pumps, we have those voltage gated calcium channels. I forgot to add that to the anatomy, so I apologize for that. But now we can get back to our story. Oops, no, hold on. I want that to stay black. So that I can do that. There we go. And back to blue. And back to blue. All right, excellent. So that muscle action potential spreads down the sarcolemma, spreads down the transverse tubule. And when it spreads down the transverse tubule, it is going to depolarize our triad. So now we understand why that's significant. If it depolarizes enough, let's say to threshold, anybody remember what we said threshold was? What's the voltage of threshold?
This is the lowest, or the lowest action potential we need to.
You are correct. It
Right.
is the critical point. It is the critical change in the membrane potential we had to get to. There it is. Allison's got it. Negative 60 millivolts. Remember, we started with the cell at a resting membrane potential of what again?
Negative 70.
Yeah, negative 70 millivolts. I keep telling you that this muscle action potential is a big positive signal. A muscle action potential is on average around a 100 millivolt change. When I say it's a big positive signal, it is a big positive signal. So if you have a 100 millivolt change spreading along this plasma membrane, do you think that's going to be able to get it to reach threshold? Yeah, absolutely, because that getting threshold is only a 10 millivolt change, and this is more than enough. Now, as we know, when you reach threshold, here in our triad, what is the immediate effect going to be?
Calcium voltage, calcium gated are open.
There you go, perfect. The immediate effect is that our voltage gated, and notice I wrote it out once so I can abbreviate it here, our voltage gated calcium channels open. Excellent. And when our voltage gated calcium channels open, what is the immediate effect of that going to be?
Let the calcium go in,
In?
out of the cell. So I mean,
Well, not, be careful. It doesn't go out of the cell, but you have the right
into
idea.
cytoplasm from endoplasmic.
Perfect. Excellent. Calcium is able to exit the sarcoplasmic reticulum and enters the cytosol. So that calcium is going to come out. We have our jailbreak. All of that calcium comes rushing out of these voltage gated calcium channels, and we get all this little bit of calcium that is now being released into the cytosol. Excellent. Now we have this pump. Is this pump going to be taking some of that calcium and pumping it back in?
Probably yes.
Yeah, absolutely. But poor little Barney Fife here isn't going to be able to keep up with all of the calcium that is rushing out of those gates. So more comes out of the sarcoplasmic reticulum than these poor little pumps can go back in. So what ends up happening is we get an increase in the concentration of calcium in the cytosol. Excellent. Now, I know my picture doesn't do the best job of showing this, but remember, the sarcoplasmic reticulum is wrapped around the myofibril. So when that calcium is released, it goes straight to the myofibril. And when it goes to that myofibril, what it is going to do is the calcium is going to bind to our troponin. There's one, and there's one. I'll do the same thing on this one here. There, and there. Excellent. So our calcium binds to the troponin. And when it binds to troponin, and again, these are always one of those fun phrases that we can call grandma and tell her on the phone, our troponin undergoes a conformational change. All right, again, during the break, call up grandma, say, hey, grandma, guess what? When calcium binds to troponin, troponin undergoes a conformational change. And she'll be really impressed and she'll send you 20 bucks in the mail. But remind me again what that means. What does it mean to undergo a conformational change?
Maybe the size of the shape is changed, or the
Yeah, it's simply that easy. The troponin changes shape. That's all it means. It's just a fancy way of saying that troponin changes its shape. And if you want to say troponin changes its shape, I am totally fine with that. Saying it undergoes a conformational change is fun, but the important thing to know is that troponin is going to change its shape. And when troponin changes its shape, what it does is it rotates the tropomyosin. So what happens here is that this troponin, when the calcium binds to it, it is going to change its shape. And when it changes its shape, it is going to rotate the tropomyosin. So let's cheat. We'll put that there. We'll put that there. We'll put the calcium on them. So put that there. It rotates the tropomyosin. And notice when the tropomyosin is rotated out of the way, now our myosin binding site, you all right? Your myosin binding site on the actin is exposed. And now that the myosin actin, the myosin binding site on our actin is exposed, then what can happen is our myosin heads can now grab onto the actin, and we can start our contractile cycle. And once myosin grabs the actin, we can start our contractile cycle.
Sir, can you repeat this portion when the rotation happens? The
Yes.
on myosin or myosin binding site on actin molecules. What?
So remember, if you remember what our actin looks like, remember our actin is that dimer, and it has a handhold on it. That handhold on the actin is a place for that. Oops, hold on, I want to make this pink. On the actin dimers, remember there is a little binding site where the myosin can grab onto it. That's how we're going to get our contraction to occur. Myosin grabs onto the actin and pulls on that actin. It can't grab onto the actin and pull on the actin when that tropomyosin is in the way. So when the tropomyosin rotates out of the way, those binding sites on the actin are now exposed, and now myosin can grab on, right? I used that example, I think we talked about it way earlier, about that ladder at McDonald's that has the plexiglass over the top. You can't climb a ladder if it has plexiglass over the top of it. But as soon as you move that plexiglass, then your hands can grab onto the rungs of the ladder and pull yourself up. And that's exactly what's happening here. Tropomyosin is in the way, but as soon as tropomyosin is out of the way, then our myosin can grab onto the handholds on the actin, their myosin binding sites, and the myosin can grab and pull. And we've reached our goal. We've used calcium, we've moved our regulatory proteins, and now our contractile cycle can begin. All right. Questions on this? All right, let me save my picture, and now let's go back to the textbook, because the textbook will do a prettier job of showing this. Excellent. Notice here we see our process, the beginning of our process of the contraction of our muscle. Again, here we have our neuron with its synaptic end bulb, with its vesicles, releasing its neurotransmitter to that motor end plate where there are the chemically gated cation channels. But notice that motor end plate is just a small specialization of our entire sarcolemma. The sarcolemma is the plasma membrane of the entire cell. So we see this big, huge, massive sarcolemma, including the invagination, that fold in the sarcolemma, that is the transverse tubule, that is associated with the terminal cisternae of the sarcoplasmic reticulum, which wrap around our myofibril. So here we see a kind of a big global view of this anatomy. And here we see more of an up close view. All right. So we've got the anatomy. Oh, and notice here we also have the voltage gated calcium channels. So that again is shown here as well. So if we are going to start a muscle to contract, we need to initiate excitation, contraction, coupling. And that begins where the initiation of the communication at the neuromuscular junction ends. We have produced a muscle action potential, and it is traveling down the sarcolemma. Now, if on the exam I ask you to describe initiation of excitation contraction coupling, do you need to tell me everything that happened at the neuromuscular junction?
No.
No, that's not part of the question. You start right here. You start with the muscle action potential traveling down the sarcolemma. That is the beginning of step 2. And as we know, not only does it travel down that specialized plasma membrane, but it travels down the special folds of the plasma membrane, where it spreads across the triad. And when you have that big positive signal spreading across the triad, it depolarizes the triad. The immediate effect, oh, there it goes. Oh, see, I didn't even have to do that. Depolarizes the terminal cisternae. Now, as we know, the immediate effect of depolarizing the terminal cisternae is that they are going to open voltage-gated calcium channels. And calcium is going to rush out of the sarcoplasmic reticulum into the cytosol. Yes, there are going to be pumps that are going to be trying to push it back into the sarcoplasmic reticulum, but it's not going to be able to keep up with the jailbreak of calcium that is being released from the sarcoplasmic reticulum. The sarcoplasmic reticulum contains a massive amount of calcium, second highest concentration in the body. In fact, there's a special protein I'll tell you about in a little bit that actually helps to store all that calcium in there. So we have this huge, massive rush of calcium coming out that those poor little pumps can't keep up with. Notice here, we see the up-close anatomy that we talked about as well. So again, here is our thick myosin filament with one of its myosin heads. Here we see in blue that dimer protein that is the actin. And those black circles on it represent those handholds for the myosin binding sites. Myosin really wants to grab onto the actin. That's all it wants to do. That's its goal in life. Grab onto the actin. But that pesky tropomyosin is in the way. And so it can't. However, in the presence of all this calcium, the calcium binds to the troponin. And when it binds to the troponin, troponin undergoes a conformational change. Our troponin changes its shape. And as our troponin changes its shape, it rotates the tropomyosin off of the binding site. And now the binding site is exposed, and now the myosin can grab onto the actin. If I remember correctly, I think this is the picture from your textbook. Oh, see, there you go. Myosin grabbing the actin, and our contractile cycle begins. I believe these are the pictures from your textbook, but I have some other pictures that I've gotten from other sources that I really like that do a good job of showing this as well. So I wanted to share these with you. They're on the slides because I think it helps to see this a couple of different ways. And here we'll talk about them here. Here again, we're seeing the entire process in a very simplified view. Again, that muscle action potential spreading down the sarcolemma, spreading down the transverse tubule, depolarizing the triad, releasing the calcium, which moves the regulatory proteins and allows the myosin and actin to interact. But here's the one, this one's my favorite. I like this because normally when we look at this, like all the pictures we've drawn so far, we're all looking at it from a longitudinal view. What's cool about this one is in this case, notice we're looking at it a cross section. So here in looking at a cross section, we see our big, thick myosin filament with all of those tail regions bound together and a single myosin head. Same thing over here. And here we see that dimer that is the actin. And here in yellow is that active site, the binding site for myosin. Myosin wants to grab that binding site, but here in green, that pesky tropomyosin is in the way. However, troponin is this handle sitting on top. When the calcium is released, calcium binds to the troponin, and troponin changes its shape and it rotates. And notice as it rotates the tropomyosin out of the way. And now the myosin head can bind onto the myosin binding site on the actin. And once it binds, our contractile cycle begins. One last pretty picture that shows this. Here is our sarcoplasmic reticulum with all that massive amount of calcium stored inside of it. Here we have the V shape, in this case, voltage-gated calcium channels, and they are closed. And what I like about this picture is it shows those calcium pumps that are trying to lock away any calcium floating around in the cytosol. When the muscle is at rest, our regulatory proteins, that tropomyosin and troponin, are in the way, and the myosin heads cannot bind onto the actin. However, when we produce that muscle action potential, it goes across the sarcolemma, down the transverse tubules, so these represent the transverse tubules, depolarizing our triad, opening the voltage-gated calcium channels, and calcium rushes out. Binds to the troponin, changes the shape of the troponin, exposing the binding site, and now the myosin heads can grab and can pull. And again, these pumps are still working, trying to put the calcium away. But there's so much calcium inside of these sarcoplasmic reticulums that when we open these gates, so much calcium comes out that those poor little pumps can't get it all back in. So we get an increase in the calcium in the cytosol. Notice the overall amount of calcium in the cell hasn't changed. It's just the location of it. Right before it was locked up in jail, now it's free to move around inside of the cell. Now, as I mentioned, the sarcoplasmic reticulum has the highest concentration of calcium outside of the matrix of the bone. And one of the reasons for that is there is a special protein inside of the sarcoplasmic reticulum known as calsequestrin. Calsequestrin is a special protein that helps to store massive amounts of calcium inside of the sarcoplasmic reticulum. It binds it, it organizes it, right? Again, if you think of it in terms of packing, if you are packing to go away for the weekend and you just randomly take clothes and throw it in there, your bag fills up pretty quickly. But if you fold things up really nicely, you roll them up tightly into a ball, you can fit a lot more stuff in there. And so this helps to organize the sarcoplasmic reticulum and get massive amounts in there. And again, as you can see, it's another one of those alphabet soup terms that you're likely going to have to spell, but there you go. Cal, of course, refers to calcium. What does it mean to sequester? Yeah, to keep away, to lock away. You hear them talk about a lot with juries. If there's a jury on a very visual case, something that's in the news, they don't want people hearing about it. They don't want people talking to them. So they sequester the jury. They lock them away. And that's basically what it means. Calsequestrin, it's an alphabet soup term, but the name tells you what it is. It sequesters, it locks away calcium. All righty. Questions on that? Excellent. So we have initiated excitation contraction coupling. Any questions on that? Because we got to move on to the next one, but I don't want to move on if this doesn't make sense. All right, excellent. So then now that we have, now that we have initiated excitation contraction coupling, our next goal now will be to terminate excitation contraction coupling. And again, Initiate means start the process. Terminate means end the process. So all I'm saying is how do we start it, which is going to lead to the muscle contraction? How do we stop it when we relax? I'm purposely using the terms initiate and terminate because those are the terms I want to use on the exam so that there's no confusion. So that the two questions that you could possibly see are how do you initiate excitation, contraction, coupling, or describe the process by which you would initiate excitation, contraction, coupling, and describe the process by which you would terminate excitation, contraction, coupling. So again, I'm using these standard words so that it is clear what we are talking about. But when we want to terminate it, basically we want to stop this process so that the muscle will relax. All right. Now, if you remember when we terminated communication at the neuromuscular junction, the end of that termination was that there was no more muscle action potential. So if we're going to terminate, we start with there being no more muscle action potentials. We're not producing any more muscle action potentials. That, of course, means that there is no big signal traveling down the sarcolemma. And if there is no big signal, no big positive signal, I guess I should have thrown positive in there, signal traveling down the sarcolemma, there's no big muscle action potential. So let's go ahead and B, no muscle action potential spreading down the T-tubule. That muscle action potential depolarized our triad. So if there's no longer a muscle action potential, what's going to happen to our triad?
Repolarized?
Exactly. Our triad repolarizes. Right? And when our triad repolarizes, what's the immediate effect of that going to be?
The calcium channels blocks.
Exactly. The immediate effect of that is our voltage-gated calcium channels close. And if our voltage-gated calcium channels close, there is no new calcium into the cytosol. So let's go ahead and put some big X's. Oops, red. Go ahead and close that, and close that, and close that, and close that. So no new calcium into our cytosol, but we still have the issue of there being a lot of calcium already in the cytosol. Do we have a way to get rid of that?
The pumps.
Yeah, now finally the pumps will be able to make a difference. So our calcium pumps remove the calcium from the cytosol. All right, they lock it back up in the sarcoplasmic reticulum. And so as they, there we go, sneak that in there. So now there is no calcium to bind to troponin. And if there's no calcium to bind to the troponin, then what happens is troponin returns to its original shape. Oops. And when our troponin returns to its original shape, guess what it does? Oops, no, didn't want that. Without the calcium, so now I got to be tricky. Grab that, move that out of the way. Grab that, move that out of the way. So that without the calcium, it is going to go back to its original shape. And when it returns to its original shape, our troponin rotates the tropomyosin back over the myosin binding site on the actin. And if we rotate that binding site, that troponin, pardon me, if we rotate the tropomyosin back over the binding site, can myosin grab onto the actin anymore?
No.
Easy questions. Yeah. Myosin can no longer bind to the actin, and that is going to stop the contractile cycle. There you go. Mission accomplished. We have terminated excitation contraction coupling. All right. Questions on that?
So when we are going to terminate the excitation coupling process, so there is no big positive signal traveling to sarcolemma and also to the transverse tubules, and which is going to be repolarized, right?
Correct.
Yeah. And the immediate action is like the calcium channel blockers. Gates are closed.
Yep.
Thank you.
Yep. Then no new calcium out. Then calcium gets pumped away with no calcium. Troponin goes back to its original shape, rotates the tropomyosin, blocking the binding site, and myosin can no longer bind to the actin. All right. Excellent. Now, if you remember when we were talking about, so questions on this, because we got one more small, tiny piece to do. All right. When talking about these processes, remember we also talked about the need for being able to understand how to modify. However, modification of this system is a little more complex than this. The short answer to how this is modified is really a major part of the process that we call fatigue, right? What does it mean for a muscle to be fatigued?
An ability to contraction.
No, see, and I think that's, I'm very glad that you said that, because I think that that is a common misperception. That is not what fatigue is. It's not stressed either. It can be caused by stress or overuse, but what it means for a muscle to be fatigued is that that muscle is not able to perform at the level you want it to, right? Last night was game 6 of the World Series. And I don't know how many of you follow baseball or any of that, but Tampa Bay's starting pitcher got pulled in the 6th inning, right? Because he was fatigued. Does that mean that he couldn't move his arm anymore? No. He was throwing the ball at the beginning of that game at 100 miles an hour. By the 6th inning, when he threw that fastball, it was only going 95 miles an hour. Guess what? Throwing a baseball 95 miles an hour is pretty damn impressive. But was it the level that he was pushing it at before?
No, the muscle.
No. So notice the muscle did not perform at the level that we wanted to. I grew up back east, right? So again, it's a totally different planet back east. And when I was in middle school and you got in trouble, what you had to do is you had to go up to the principal's office. And when you went up to the principal's office, he would have you put your arms out, palms up. And he would put a textbook in each of your hands. And you had to get down on your knees and you had to hold up those textbooks until he got tired. If you could hold them up until he got tired, then you got to go back to the classroom. If you could not hold them up till he got tired, then you got swatted and sent back to the classroom, right? And all the times I got sent to the office, was I ever able to hold those books up till he got tired? No. Eventually my arms fatigued and my arms dropped and I dropped those books. Does that mean I couldn't move my arms anymore? No. It just meant that I couldn't perform at the level that I wanted to. Yes, actually, one of the Dodgers players did test positive, or his positive test for his COVID test came back positive during the middle of the game. He got pulled from the game, but that didn't stop him from running around without a mask and celebrating at the end of the World Series. So go Dodgers. There you go. So yes, so again, fatigue means not being able to perform at the level required. Right? He wanted to throw at 100 miles an hour. He couldn't. I wanted to hold those books up and I couldn't. The muscle didn't fail in that I could still move my arm, right? I got back to the classroom and I was able to write in the books just fine, but it couldn't perform at the level that I wanted. One of the main factors of that fatigue, the whole reason I went on all this what seems like a tangent story, is because one of the factors that influences fatigue, and we'll talk about fatigue in more detail in a couple lectures, is a change in pH. Why would the pH inside of a muscle cell change?
Because of other substances like lactic acids.
Yeah. As we're metabolizing, we can produce things like lactic acids. And as we know, pH, let me say, oh, not only do we change the pH, but when we use a muscle, we also generate heat, right? So when you use a muscle, you generate heat. When you use a muscle, it changes the pH of the muscle cell. And wasn't there something that we said, a macromolecule that was highly sensitive to pH and temperature changes? What macromolecules are highly sensitive to pH and temperature changes?
Nucleotides, the protein.
No, it wasn't the nucleotides. It was that second one. How about proteins?
Proteins, yeah.
Yeah, proteins are highly sensitive to pH and temperature change. And remind me again what a muscle cell is just chock filled with? Proteins. The proteins in here can be affected by that pH and temperature. And one of the biggest ones that is affected by pH and temperature changes is the troponin. When the pH and the temperature of the cell changes, what happens with troponin is troponin changes its shape just slightly. And that minor change in its shape makes it harder for the calcium to bind to it. So notice, everything else can go perfectly. You generate your muscle action potential, you depolarize your triad, you open the calcium channels, you dump a bucket worth of calcium into the muscle cell. But if that calcium can't bind to the troponin, it can't change the shape of the troponin. It can't move the regulatory proteins. And if you can't move the regulatory proteins, myosin and actin can't interact, and suddenly the strength of your contraction goes down. So what I would say is when we talked about those 10 possible essay questions, modifying excitation, contraction, coupling isn't going to be one of them. We will talk about fatigue. We will talk about some other factors there, but that will be a different type of essay question. So for part 2, excitation, contraction, coupling, you are just going to have to worry about initiation and termination. All right. Questions on that? And look, now you only have to memorize 9 essay questions. Getting easier by the second. All right. And I'll tell you a secret. You're only going to have to memorize 7. So we're going to lose 2 more as well as we go all the way. All right. Questions on that? All right. Excellent. With that, then, we are done with part 2. So we only have one more part to go. Part 3, contractile cycle. So let's go ahead. This is a good stopping point. Take our first break. Any questions on this process before we take our first break? All right, excellent. Then it looks like it's 9:14. Let's come back at 9:29. And at 9:29, we will pick up the lecture from here. So let's pick a color we haven't used yet, purple. 9:29, restart. And I'll start the recording at that point as well. All right. See you guys. Here, I'll stick that up there. Excellent. See you guys in 15 minutes. All righty, let's go ahead and get started. So I know we just went over this, but I think these overviews are useful and helpful. So we've done the second one. Let's move on to the third step. Someone remind me again what step 2 was, though?
Excitation, contraction.
Excitation, contraction, coupling. The goal?
To use calcium and remove the irregularity proteins.
Excellent. What was the, okay, let's be cheap. Part 2. Excellent. What was the first step in excitation, contraction, coupling?
Start of action potential and the motor end plate.
Motor end plate, I like that. Spreads down the sarcolemma. Excellent. And what was the last step?
Termination. Termination of?
No, not for termination. So, okay, well, although we want to be careful, Allison, myosin binding to the actin is really part of the contractile cycle. So what allowed the myosin to bind to the actin? What is it allowed that interaction to begin?
Troponin?
The myosin binding site was exposed on the actin.
Excellent. Exactly. So our troponin changed shape and rotated the tropomyosin, which exposed the myosin binding site on the actin. Excellent. And again, it's useful to know what the last step of part 2 is, because how does that relate to part 3?
Because the part 3 starts from that.
Exactly. It's going to be the first step of part 3. So troponin changes shape and rotates tropomyosin, which exposes the myosin binding site on actin. Excellent. And what is part 3 again?
Contractile, or we can say sliding filament theory.
Excellent. Contractile cycle, or we can also call it the sliding filament theory. And actually, let's not even put in parentheses. Let's just put a slash there because both terms are equally acceptable. What's the goal of the contractile cycle?
To generate force or tension and make the movements.
Force or tension, depending on how you want to think of it, to change the shape, of the muscle cell. Excellent. Perfect. So there you go. So we have summed up part 2, gotten a good head start on point 3. Part 3, our contractile cycle and what is involved in that. All right, questions on that? All right, excellent. So just like we've done with the previous 2 processes, this is going to need to begin with some discussion of anatomy. So let's go ahead and clear this. Give me this up here at the top. So we are now on to part 3, our contractile cycle. And again, our goal here is to generate tension or force and change the shape of the muscle cell. Excellent. All right. Now, again, this occurs at the sarcomere. So since this occurs at the level of our sarcomere, we need to look at the anatomy of the resting sarcomere. Most of this is stuff that we've already talked about and we kind of already know. So we need to know the anatomy of the resting sarcomere. Of course, as we've talked about, what forms the boundaries of the sarcomere?
Z discs.
Z disc, absolutely. So we have that Z disc, that zigzag protein located on the boundaries. And again, remember, these are the walls. So what I've got here from Z disc to Z disc is one sarcomere, and then there's a second one going off that way and a third one going off that way, and so on and so forth. We also have in the center that big, huge, thick protein. That is our myosin. As we know, we have the myosin heads coming off of this, and I'm going to draw 8 of them here. And this time I will draw the actual head unit for it. And remember, as we've talked about, these myosin heads are motors. What does it mean to be a motor again?
Moving.
Say again?
Moving.
Okay, true. So it's a little more than that, though. What does it mean to be a motor? What is the job of a motor?
Generates energy or force.
Excellent. Perfect. I like that. So it provides a mechanical force. However, what does it provide that mechanical force from?
From chemical force.
Exactly. So what it does is it uses a chemical force to provide a mechanical force. So really, the other way you could say this, and again, I'm fine with either way, it converts chemical energy into mechanical energy. And so that is, oops, if I spell energy right, what our myosin heads do. Our myosin heads are these motors that are going to convert our chemical energy into mechanical energy. Right? In this case, absolutely, the chemical energy uses ATP. It rips that phosphate off of the ATP using the energy released from that high energy bond to change its shape and do work. Absolutely. And when you rip that phosphate off of ATP, you get a DP and a massive amount of energy. Absolutely. Excellent. Of course, if it's going to convert that and do that, use that motor, may change its shape, it has to pull on something. And that's something it's going to pull on is, again, the actin. And I will exaggerate the space between them a little bit again there to give us some playroom. let's go that there. Let's go that there. And I need to cheat and go, whoops. Come on. Thank you. Perfect. Excellent. So we have that there. Our actin. We also know this time, oh, so I need to move this here and do that so that I can put a second of our structural proteins, and that is that myomecin. Remember that myomecin is those 3 lines of interlocking proteins that stabilize the myosin, hold the myosin to the other myosins, because remember there's going to be another myosin up here in this myofibril, and it also goes across from this myofibril to the next myofibril, helping to line them all up perfectly with each other. So we get that perfect overlap. Sorry, hold on. I apologize. Excellent. All right. So that myomecin perfectly aligns all of these things together. So we have that anatomy to them there. We know we also have that titin, which is going to stabilize, and we'll just go ahead and draw that as a reminder that it's here. Again, it's not going to really play a role in this process, but since we're drawing all of our proteins and the entire anatomy of our sarcomere, it's worthwhile to have it because, of course, having you describe all of the proteins of a sarcomere is certainly a potential essay question. That titin, remember, provides the elasticity and the extensibility of our muscle cell, allowing it to be stretched without damage and allowing it to recoil to its original shape. It also helps to stabilize the myosin there in the middle. And as we also talked about, we know we have, and I'll change colors of them this time, those regulatory proteins. So we have, oh, no, I don't want that then. Here we go. That thread-like tropomyosin that is blocking the binding sites on the actin, and that handle-like troponin that is going to help us to move it. Notice there really isn't too much new that we've added from the previous picture when we draw this, but it's always good to make sure we understand these things. Let's talk about the myosin. So let's go ahead and label all these things so that we have it to remember the stuff by. And this is our actin, and this is our troponin, and this is our tropomyosin. And this is our titin. And this is our myosin. Z-disc. And myomecin. I think that's everything. I labeled everything. 1, 2, 3, 4, 5, 6, 7. That looks right. 1, 2, 3, 4, 5, 6, 7. Yeah, okay, excellent. Those are the 7 proteins we are responsible for. Now, the other reason that I want to take us and draw this. So again, really nothing too new based on what we've talked about so far. However, in this resting sarcomere, there is one more thing that I want to point out to you. Notice I've done it twice now, and again, it has been on purpose, so I want to make sure to emphasize this. Notice that at rest, the myosin heads are pointing towards the Z-disc and away from the M line. So when the muscle cell, so let's go ahead and change colors of this just to emphasize it, that when the muscle is at rest, the myosin heads are pointed away from the M line and towards the Z disc. So that's really the only other anatomical thing we need to talk about, except for the other nice thing about all these proteins is these proteins are so precisely arranged that because of this precise arrangement, they actually give our muscle cell stripes. So for instance, the region of the, oops, didn't want that, I want this. The region of the muscle cell that all the myofibrils contain myosin in them, what do we call this region? The region of the organelle that just contains myosin in it is the dark stripe. Of course, we're not going to call it the dark stripe.
I bond.
Is it the I band? The dark stripe?
A bond, sir.
A band, excellent, A band, excellent. Whereas the portion, and I guess for this we have to cheat and draw a couple more myosins just barely peeking out here. So we'll take a myosin there, and we'll take a myosin there. Excellent. So then the portion of the myofibril that doesn't contain myosin, what is that part?
I band.
I band, excellent. In the middle of the I band, oh, that would be nice if I capitalized it, there is the Z disc right at the center, which as we know can be seen in the light microscopy, and that is our Z line. And smack dab at the center where the myomecin and the myosin overlap, since it's both myomecin and myosin, what do we call that area right at the center? Excellent, that is our M line. And then there's just one more region we need to be able to identify. Again, the A band is this entire region that contains the myosin. That is our A band. However, if you notice within the A band, there is some portion of the A band where actin and myosin overlap. But there is also a smaller portion of, oops, and that line there was horrible. So let's go back. A smaller portion of the A band where just myosin is located. There's no actin. And this part in the middle that just has myosin in it, what did we call that region?
H band.
H band.
Or the H zone, excellent. H band or the H zone, both are acceptable. Perfect. So now with this, we have been able to identify all of the proteins that form the sarcomere. We have been able to see how they're oriented and related to each other, and we see how they make up the stripes. All right, any questions on any of this? All right, this is our anatomy for our starting point. However, this takes up a lot of space. So to really understand what this is going to do and how we're going to do it, we're going to cheat a little bit. What I'm going to do is I am just going to redraw basically this. I'm going to take 1/4 of this sarcomere. And we're going to talk about what happens just in this one small segment. Now, is what's happening here at the top the exact same thing that's happening here on the bottom? Absolutely. Is what's happening here on the right the exact same thing that's happening on the left? Absolutely. So even though we're looking at a small part of it, we know this is going to be taking place in the entire sarcomere. But by focusing just on this one small quadrant, it will make it easier to draw, easier to talk about, easier to understand. All right. So to do that, I have to redraw my picture. So before I erase this, before we move on, I want to make sure we understand the anatomy. Is there any question on the anatomy before we get started? All right, perfect. So then in that case, what we're going to do is go ahead and I'll go ahead and save that. Clear my drawing. And now we have to write again. What we're doing. Part 3. Contractile cycle. Why the first C isn't. Contractile cycle. And again, the goal of this is to generate force, generate that tension, and that is going to allow us to change the shape of the muscle cell. All right, so that's our goal. That's our starting point. Now, as I mentioned, we need to draw our basic starting anatomy. So for that, and we'll do this right here, we have our myosin, we have our actin, and I'm just going to draw one actin head. I don't like that. Bigger. Thank you. One actin. Oh, hold on. I need to move this a little bit. You come down. You come down. And we will draw the Z disc over here and draw our actin up here. Shorten that. I need room to play, so I apologize. It's taking me a little longer to get this set up, but perfect, excellent. And then our nice big myosin head. Excellent. Now, one more thing we need to know about this myosin head. Remember, as we talked about, when this myosin head is at rest, it is pointed away from the M line. And at this point, what we have is, the reason it's pointed away from the M line is because it is a primed myosin head. What that means is that this myosin head has already split an ATP. And remind me again, when you split an ATP, what do you get as a result of splitting that ATP?
ADP.
You get ADP, you get a phosphate, and you get a tremendous amount of energy from breaking that bond. All right, so those are the things that are here. However, at this point, the myosin head can't use that energy for anything. So at this point, it's actually still holding on to all of it. In this myosin head, it has the ADP, it has the phosphate, it has the energy, but it can't do anything with these things yet because it can't bind onto the actin. So in this case, it is primed. It is split the ATP. It is holding all of these things. It can't release it yet. And when it's in this prime position, as we mentioned, it is pointing away from the M line. So this is our primed starting point. All right. Now, if we are going to start our contractile cycle, what's the first thing that has to happen? Hold on one second.
Binding of the troponin to the actin.
Excellent. So what's our starting point again?
Troponin with the actin, tuple actin.
Excellent. So absolutely correct. Whoops, wrong button. This one. So remember, our starting point is that our calcium is bound to the troponin, changes the shape of the troponin. So our troponin changes shape and rotates the tropomyosin. And when it does that, it exposes the myosin binding site on the actin. On the actin. So this is our starting point, right? So again, our tropomyosin is going to get rotated out of the way by that troponin that has, and let's put some fun little yellow calciums on there. There's a calcium and there's a calcium. So it is moved out of the way. And when it's moved out of the way, now finally that myosin head is able to grab onto the actin. And that is what's going to happen. So we need some more anatomy, because what is going to happen, the first thing that is going to happen, cheat and move that there, cheat and move that there. Our myosin is going to grab onto our actin. So we have our myosin there. We have our Z disc here. We have our actin here. This a little more this way. This way. And then our myosin head is able to bind onto the actin. Excellent. So our myosin head binds to the actin. Now, that's a simple enough event, but if we know anything about our anatomists, they love to name everything. So we have a fancy name for this process. This fancy name of the process of the myosin head binding to the actin is, we say it forms a cross bridge. So our myosin head, or primed, let's be more specific. Primed myosin head forms a cross bridge. And that forming of the cross bridge is, just like we said, the myosin head binds to the actin. And again, remember, it still has the ADP, the phosphate, and the energy held inside of that myosin head. So the first thing that happens as soon as those regulatory proteins are out of the way is that our myosin head is finally able to grab onto that handle of the actin. And now that it's formed that cross bridge, now it's going to be able to finally release that energy. And let's actually say it that way. Release the energy, use the energy to do work. And so that is what's going to happen next. As soon as it binds onto that actin, then it is going to be able to finally do some work. That process of doing the work is what we call, again, because they like their fancy terms, performing a power stroke. So our myosin head will perform a power stroke. What this means is that our myosin head, and so let's go ahead, we got to draw our art again. There's our myosin filament. Here is our Z disc. Here is our actin. But what it means to perform a power stroke is that the myosin head pivots. And And as it pivots, it rotates towards the M line. And actually, you know what? I'm going to put this over here. I want this over here. So our myosin head pivots and it rotates towards the M line. So our myosin head comes this way. So again, we have this pivoting towards the M line. Now, notice as it pivots towards the M line, it is going to be pulling the actin towards the M line. And because it's pulling the actin towards the M line and the actin is connected to the Z disc, it pulls the Z disc towards the M line. Now remember, this is happening on both sides. So over here on the right, it is grabbing and pulling, and the Z disc is moving this way. And on the left, it is grabbing and pulling. So the Z disc is moving this way. So notice as those myosin heads grab and pull, both Z discs are pulled towards the M line. And if both Z discs are pulled towards the M line, what happens to my sarcomere? It gets shorter, absolutely. My sarcomere gets shorter. All right, so far so good. My sarcomere is getting shorter, perfect. But that means if my sarcomere is getting shorter, my myofibril is getting shorter. My myofibril is getting shorter. Remember, there are more structural proteins that hold the myofibril to the sarcolemma. So that when the myofibril gets shorter, it pulls on the sarcolemma and my muscle cell gets shorter. Notice, we've achieved our goal. Our goal was to produce tension and change the shape of the muscle cell. We have officially, gold star, reached and accomplished our goal. Mission accomplished. Story over. Okay, maybe not. But we have made the muscle shorter. We have generated our tension. We have changed the shape of our muscle cell. But is one myosin head grabbing and pulling one time going to be enough to accomplish our goal? If I'm trying to grab a ladder, do I grab one rung, pull myself up that one rung, and then call it a day? No, absolutely not. It's not over till I get to the top of that ladder. And the same thing is true here. While we have changed the shape, while we have generated the tension, remember, this is a cycle. We want this to be a continuous process. And so for this to be a continuous process, we need to continue to not just grab that handle and pull once, but to continue to grab handles and continue to climb that ladder. Now, there's one other important thing that happens here when this myosin head performs its power stroke. During the power stroke, the myosin head obviously uses the energy from splitting that ATP, right? Obviously, that's where the energy for this power stroke is coming from. But the other thing that happens during the power stroke is that the myosin head releases the ADP and the phosphate. So during this power stroke, the ADP and the phosphate are released from the myosin head. And that means that our myosin head has a free binding site or a new ATP. All right. Questions on that? All right. I love using the analogy of grabbing a ladder because that is very much what this process is like, or pulling on a rope, right? If you think about if I'm climbing that ladder, the first thing I have to do is I have to prime my arm. I have to get my arm up there and ready. As soon as it's up there and ready, I need to grab onto the rung, right? Form that cross bridge. As soon as I form that cross bridge, I then have to use my force, use my energy to pull myself up. And once I do that, if I want to keep climbing the ladder, what do I need to do next? I've reached, I've grabbed, I've pulled, right? Any of you climbed a ladder before? And if not, please go out into your garage and do so. Once I reach, grab, pull with my hand, what's the next thing I have to do with my hand if I want to continue the process?
Let go and grab again.
Exactly. I need to let go. The very next thing I have to do is let go so I can start the process again. And that's a problem because notice I've used all of my energy and that myosin head can't let go by itself. Luckily, when it released that ADP and that phosphate, it now has a new open binding site. And that new open binding site is going to help me to be able to let go of that rung. So again, we need our myosin here. We need our Z disc. We need our actin. And we need our myosin head. Now, as I mentioned, that myosin head now has a free open binding site. And so what happens is we now need to break the cross bridge. The way this occurs is that a new ATP binds to the myosin head. Period. It's not using the energy from this ATP. What has happened is that a new ATP has come in and bound to that binding site. So a new ATP binds that open binding site on the myosin head. And just the binding of the new ATP causes the myosin head to change its shape, undergo a conformational change. And that change in shape, it releases the actin. Now notice the binding site on the actin is free, and our myosin head has let go. However, at this point, is our myosin head ready to do work? Is it in a position where it looks primed and ready to do work? Right, exactly. However, the myosin head is not ready to do work. To be able to do work, the myosin head must become primed. And luckily, we know how to become primed. How do you prime a myosin head?
Remove the phosphate.
Right, split the ATP. Right, so our ATP is split into ADP, phosphate, and energy. This rotates the myosin head into the primed position. And now it is ready to grab again if the binding site is open. Which brings us right back to where we started. Notice again, we said this is a contractile cycle because it is a continuous process. Just like me climbing the ladder, I reach, I grab, I pull, I let go. I reach, I grab, I pull, I let go. If I'm climbing the ladder, there are 4 steps in that process. Reach, grab, pull, let go. Reach, grab, pull, let go. And that is exactly what our myosin head is doing. Our myosin head reaches, right? This is the reach. This is the grab. This is the pull. This is the let go. And then, of course, once you let go, you can reach, grab, pull again. And so we have this continuous process where the sarcomere continues to get shorter and shorter and shorter. We generate more and more and more tension, and we move that muscle through space.
One question, sir.
Yes.
If this process is going like by splitting ATP to ADP, phosphate and energy, and also by rotating the myosin head to the prime position. So if the site is not ready, the actin site is not ready, what will happen?
That is an excellent question. Now let's take it a step further. You may not have thought of it in these terms, but most of you are probably aware of this process because, as you very pointedly mentioned, to continue this process, we need ATP. Luckily, the cell always has ATP in it. However, would there be a condition where the cell wouldn't have ATP in it? Could there be any potential moment in a cell where it would not have ATP, would not be producing ATP?
Probably yes.
Yep. And what would that condition of that cell be? What do you think?
Rest.
What do you think? Rest? Well, okay. But remember, we have lots of pumps. We have lots of other things. Again, what is the condition of your body if you are not making any ATP in any of your cells?
Dead.
Dead. There you go. Absolutely. You are dead. Not starving, right? Not sick or anything else. Dead. So here you are wandering around the street, right? And there you see that dead body on the side of the road, right? And when you see that dead body on the side of the road, right, often when the EMTs come to pick them up, right, there you go. They call them stiffs. Why do they call them stiffs? Because they're locked in the position that they were in when they died. Why does that occur? Because what happens is your muscles, when you die, stop producing ATP. That ATP is normally being used for things like pumps to keep the calcium out of your cytosol. But without ATP, when you're dead, calcium is going to leak into the cells. And when calcium leaks into the muscle cells, it's going to move the regulatory protein. And our myosin head is going to grab and it is going to pull. But the problem is with no ATP, it can't let go. And all of your myosin heads are locked in that position, which means your muscle is locked in that position. And as Allison pointed out, what's the fancy term they use for that when the body's locked in that position? Rigor mortis. that rigor mortis that happens to the body is due to the fact that there's no ATP inside of that muscle cell, so the myosin head can't let go and it's locked in place. Now, when someone dies, do they instantly go into rigor mortis? No, it takes a little bit of time to use up the ATP. It takes a little bit of time for that calcium to sneak its way in and move those regulatory proteins. And then once the body's rigid, does it stay like that forever?
Lividity.
Yeah, eventually it goes away. And the reason it goes away is the body starts to deteriorate. The myosin heads, the actin, the Z disc, all the proteins start to break down. And as those proteins start to break down, the body becomes flaccid again. Right? One of the ways they can kind of tell how long a body has been dead is by that state of lividity. Is it, totally flaccid? Is it totally rigid? Is it partially rigid? Right? They can get some sense of how long the body has been dead for by the state of decomposition. Because first it's just dead, then it becomes rigid, then it starts to relax again. So there you go. So you may not have thought of the contractile cycle or the effect of ATP here, but most people have heard or seen rigor mortis, or at least seen some funny video or movie where they make fun of it or something like that. The person dies in some weird shape and they have to pick them up and carry them off that way. Well, this is why. All right. Excellent. We've done it here on the board. Let's take a look at it with the pretty pictures from the textbook. Weekend at Bernie's, exactly. Although I don't know if he ever went into rigor mortis. It's been too long since I've seen that movie. I'm gonna have to go back and look at it again. There you go. Excellent. All right. So again, here is that anatomy for the initiation of muscle action potential here at the neuromuscular junction. Then we have our excitation, contraction, coupling. And now our job is to talk about the contractile cycle. Notice here is a pretty picture that shows most of the proteins we're concerned about in our sarcomere. Notice it does a nice job of showing us our A band, our I band. Here we also have our H zone, where there's just myosin, our M line, our Z line. Notice this one doesn't show the regulatory proteins, but it does a nice job of showing us that resting sarcomere. And notice, as we talked about, the myosin heads on the left point to the left. The myosin heads on the right point to the right. They're in that primed position. This is a very simple illustration. Here's a little bit of a closer view of what this looks like. Like us, they chopped out a piece on the right-hand side so that now we see this resting sarcomere. Notice our myosin heads are primed, pointed away from the M line. They've split the ATP into ADP and phosphate. They have that stored energy, but they can't use that stored energy yet because that pesky troponin is holding the tropomyosin in the way. So our troponin and tropomyosin are covering the binding sites. Our myosin head has split that ATP into ADP and phosphate. It's stored that energy. It's pointed away from the M line. And again, we have a fancy term for this. We say it is primed, right? Or we can say that it's cocked, right? It is prepared to do work. We have that primed myosin head. You cock it like a pistol where it is ready to do work. Okay. So contractile cycle begins where excitation, contraction, coupling ends. And that is with the movement of the regulatory proteins. Our calcium binds to the troponin. Our troponin rotates the tropomyosin, and that active site is revealed. And now that those regulatory proteins are out of the way, now that primed myosin head can finally do what it wants to do, grab onto that actin. Of course, we have a fancy name for that grabbing of the actin. It forms a cross bridge. So here we see a couple primed myosin heads binding to the actin, forming that cross bridge. So that energized, primed, cocked, whatever term tickles your fancy, you're welcome to use. Myosin head binds to the actin, forms that cross bridge. Again, it is in that prime position where it is pointed away from the M line. And again, its orientation is important because once it binds, it is able to finally release that energy. And it releases that energy by performing its power stroke. So now our prime myosin head can finally release the energy. And it does that by pivoting or rotating towards the M line. Again, we call this its power stroke. When it's doing this, oops, let's go back. When it is doing this, again, we can walk the steps out. It pulls on the actin, which pulls on the Z disc, which shortens the sarcomere, which generates our tension, and again, it shortens the muscle cell. So we've generated that tension, we've shortened the muscle cell, we have accomplished our goal. Of course, we want to do more than one power stroke, so we have to keep the process going. And one of the ways we're able to do that is during this power stroke, remember, our myosin head doesn't just use the energy, but it also releases the ADP and the phosphate. So now it has an open binding site. And what's going to bind to that open binding site? ATP, exactly. Excellent. So that new myosin head binds to the ATP and it breaks the cross bridge. Remember, that's all it does. It breaks the link between the myosin and the actin. So our active site on the actin is exposed again. But remember, we still need to prime the myosin head like we see here. So that myosin head has to split the ATP into ADP and phosphate. It reprimes or recocks the head, points it away from the M line, and ready for the process to begin. Notice the picture from your textbook doesn't really show that breaking of the cross bridge part, but what is nice, I do like this picture. Again, this one's not from your textbook, but this is the one that I base my drawing off of, and I have it on your lecture slides because I think it is useful to see, where again, we have those 4 steps in the process, right? It starts with a primed myosin head. That prime myosin head has split the ATP into ADP and phosphate. It is able to, yeah, let me finish this and then I'll go back, Allison. It is able to bind to the actin, form that cross bridge. Once it forms that cross bridge, it performs its power stroke, pulling on the actin, pulling on the Z disc, shortening the sarcomere, generating our tension, and releasing the ADP and phosphate. Its binding site is open, so a new ATP can come in and break the cross bridge. But notice when it breaks the cross bridge, while the actin site is open, our myosin head isn't ready to do work yet. It first has to split that ATP, and that primes or cocks the myosin head so that the process can continue. All right, I can go back to that one there. All right. Got it? Oh, well, detachment of the cross bridge. So this is again where the picture doesn't show it. The picture, your picture in your textbook doesn't show an unprimed where it breaks the cross bridge. But remember, the key to this step is that we are breaking the cross bridge by binding the ATP to it. But remember, binding the ATP, you're not splitting the ATP, you're not using the energy. It's just the binding of the ATP that breaks the cross bridge. So the ATP breaks the cross bridge, but then it has to split the ATP so that it's primed. Notice they've already shown the prime head here, but we know there's that additional step in between. again, the limitations of the artists from your textbook. I try to use the pictures from your textbook as much as possible because it makes it easier, I think, to relate to the material when you're studying on your own. But again, the problem with that is we're left with the idiosyncrasies of the artist for that particular book. All right. So did all of that, bingo. And again, here we see those 4 steps. And again, remember 5 because it started with the resting. So that's why there was 5 on the other and 4 here because it just shows the resting. All right. Now, a couple more quick things. Now, obviously, a myosin filament has hundreds, if not thousands, of myosin heads. So are all the myosin heads reaching at the same time, grabbing at the same time, and pulling at the same time? No, they're not, right? Because if you did that on a ladder, reach at the same time, pulled at the same time, grabbed at the same time, pulled at the same time, then you'd have to let go at the same time. And if you let go at the same time, what happens? You fall down. So the same way that when you climb a ladder, one's grabbing while the other's pulling or letting go, you're alternating the processes as you go through this. Laura, I don't care whether you do 4 steps or 5 steps. The 5 steps that you saw before, step 1 was the resting condition. So it started with the resting condition and then move the regulatory proteins and then reach, grab, pull, let go. So those are really the 4 steps. Their 5th that they had was the resting state. So it wasn't the resting state, was just your starting point of the cycle with that. But again, I don't care how many steps you use to describe this as long as you have all the correct information. So,
Question.
yes.
So you were saying it's like a ladder. If you let go, you fall down. So if you're going up the ladder, what makes it go back down the ladder? Do they all let go at the same time to go back down?
Exactly. The way we would stop this process is that when the calcium goes away, let's use yellow to use the same color. When the calcium goes away, the troponin goes back to its original shape, and the tropomyosin is going to rotate in front of the binding site. So now your myosin head grabs, right, reaches, grabs, pulls, lets go, it reaches, but with that tropomyosin in the way, can it grab again?
No.
No. And so as soon as the calcium goes away, as soon as that tropomyosin moves in the way again, that stops the cycle.
So now someone's poured grease on the ladder and you fall down.
Yeah, or covered the ladder up, covered the rungs up with a plastic so that basically none of the myosin heads can grab anymore, and so the muscle relaxes. All right, and like getting up a ladder, grabbing and pulling once doesn't get you to the top. Every single myosin head grabs and pulls on average about 5 times a second. So reach, grab, pull, let go. It does that 5 times in a second. And every single power stroke of those myosin heads basically shortens the muscle about 0.5%. So when I'm lifting something even as light as this pen, this flexing of my arm holding the pen, right, is all those myosin heads grabbing and pulling, hundreds of myosin heads, thousands of myosin heads, all grabbing and pulling 5 times a second to bring my arm up and lift this pen. And as we talked about, it will continue to lift, continue to contract until what happens? What are the 2 things that could stop this process?
Death.
Excellent. And why does death stop the process? You are correct. Absolutely, death would be an example. But what is it specifically about the death that stops this process?
No more calcium and ATP?
No more ATP, right? The key is no more ATP. If you think about it, calcium is not really used up in this process. It's the key that unlocks the troponin, moves it out of the way, but it's not used up. ATP is used up. Is it possible to use up all of the ATP in your body without dying? Or enough of it to be debilitating? Yeah, but it takes some pretty extreme activity. I don't know if you guys remember, in one of the Summer Olympics a few years ago, there was a female marathon runner who, again, 26.2 miles. She had gotten back to the stadium. She had gotten back to the track where they just have to do that final lap to come around. And she had so physically exhausted and depleted all of her energy and resources that she actually collapsed right there on the track. She could see the finish line from where she was, but she was so exhausted and so depleted, they basically had to come and grab the ambulance and pick her up and take her away. And she wasn't able to physically complete that marathon because she had exhausted her resources, basically depleted her ability to produce ATP and other things so much that she basically collapsed. However, again, it takes those types of incredibly extreme situations for something like that to occur. For the most part, you stop producing ATP when you die, right? So again, ATP is one of the things that keeps it going. We must have ATP to keep this going. And as you already mentioned, the other thing we have to keep this going is calcium. Notice it's the calcium not getting used up. It's calcium, or more specifically, calcium in the cytosol, right? So as we close those voltage-gated calcium channels, as we put the calcium back into the sarcoplasmic reticulum, there's no calcium in the cytosol. The regulatory proteins move back in the way, and the muscle contraction stops.
Sir, as you given the example of the marathon runner that she depleted the ATP, so can we call this condition fatigue?
I'm not certain if you would call it that. I'd call it a bummer, because it isn't just that she ran out of ATP. She also, had inflammation and so much lactic acid and everything else. So it was, I'm oversimplifying a little bit by that she ran out of ATP. She fatigued to the point of failure. So absolutely, she fatigued to the point of failure in that case. And part of that was completely depleting all of her fuel resources to make ATP. So yes. So in that case, but I'm not sure I'd go that far.
Okay.
All right. Questions on this? So as long as there's calcium in the cytosol, as long as we have ATP, this process will continue. So notice, let's go back to our picture. This would be, I guess, what we could think of as initiation of the contractile cycle. How would we stop the contractile cycle? How do you stop the contractile cycle?
Stop releasing calcium. So stop having the signal.
Right. You put the calcium away. You lock the calcium up in the sarcoplasmic reticulum. It rotates the tropomyosin back in the way, and it stops. There you go. One sentence explanation of how you would stop that. Does that seem like the kind of thing that I would ask for an essay question? Are there many one-sentence essay questions? I will be honest, in grading essay questions, I often see answers from students that are one sentence long, but are those usually getting most of the points?
No.
No. I'm looking for descriptions of things. So for contractile cycle, we're not going to worry about terminating it because it's easy. Really, the end of excitation contraction coupling termination, when you move the regulatory proteins back in the way, that ends the contractile cycle. And we're not going to worry about modifying this either. There are ways to modify it. Like I said, we talked about rigor mortis and things like that, but we'll talk about those and other things like fatigue. So for this, you only have to know the contractile cycle. All right. How are we on time? We are doing good. Okay. Questions on this? Yep. I believe Joed is correct. And I think now is a good time to take our second break. However, here's what we're going to do. I have one last physiological process I want to talk about. And then after that, we are going to go straight into the lab because, again, it's not enough for a whole hour that we're going to be talking on this. So I want to go straight into the lab. So we will take this break, but during this break, grab your handouts, grab your red and your blue pens so you're ready to dive into the lab stuff as soon as we are done with the physiology. All right. Questions on that? All right. Well, there you go. Just that easily, we have almost finished our longest physiological process. We have gotten all the way from when we decided to now we're generating tension and moving our body through space. So we have accomplished a lot in these past couple days. Excellent. So let's take one more break. Let's see, it's 10:33 right now. So let's come back at 10:48. And I will start the recording at that point. All righty, and let's make that green and bigger. Perfect. See you guys in 15 minutes. All righty. Any questions before we get started? All right, excellent. So if you've been keeping track like I have, we've been talking about the number of potential essay questions for this exam. And what would those be again? I told you we're definitely going to have at least 2 essay questions in relation to this contraction. So let's list them. What would they be?
Initiation and termination of the communication at the neuromuscular junction.
Okay, although be careful, they wouldn't be together, so they would be individual questions. So you've got the right idea though. Initiation of communication at the neuromuscular junction. And I'm going to abbreviate these in the interest of time. Then also termination of communication at the neuromuscular junction. What else? Modification, excellent. We also talked about modification of the communication at the neuromuscular junction. Now for that one, we can kind of put an asterisk next to it because I probably wouldn't ask you how do you modify communication at the neuromuscular junction. I'd more likely ask you what does the tetanus toxin do or what does curare do at, to modify communication at the neuromuscular junction. But we would talk about modification of the communication at the neuromuscular junction. What else?
Initiation of step 2, the excitation contraction.
Excellent. Excitation, contraction, contraction coupling. And again, I'm just abbreviating this for speed. What else? Termination, excellent. The excitation, contraction coupling. What else?
Nothing.
No, not nothing.
The modification is not part of the.
True. So modification isn't one of the questions, but there are still other questions. What's left? Contractile cycle. Excellent. Do we really need to worry about terminating contractile cycle or modifying contractile cycle? No, because the contractile cycle is basically the contractile cycle. So what was the 7th? I did say 7. What's the last remaining possible essay question of which you will definitely get 2 and possibly 3 on this next exam?
Compare processes.
Comparing's not a bad guess, although these don't really lend themselves very well to comparing because they're not very similar. There you go. Remember, as we talked about, if there was a 4th step in this process, that 4th step would be relaxation of the muscle. Now, if you think about it, this really isn't a new essay question. Really, if you think about this, this is basically 2 and 5 combined. So essay question 7 really isn't a new essay question. It is really just a combination of process 2 and process 5, but it is probably worthwhile to do it as a essay. So let's do that. If on the exam, and this is a good way to practice your essay writing abilities. If I asked you to describe the process of relaxation of a muscle, walk me through the steps. I'm not going to bother numbering them.
Make a decision.
Excellent. Starts with we make the decision. When we make the decision to relax the muscle, what happens?
No more NAP.
Excellent. Stop producing neural action potentials. And again, remember, we want to be careful about our abbreviations. If you're going to use abbreviations, you either need to make a key at the bottom or you need to write them out once, and then you can abbreviate it after that. So no new neural action potentials. What happens next?
Closure of the calcium, cation.
You
Chemical.
are correct.
Voltage
The calcium channels are going to close, but something's got to happen first before that occurs. Was somebody else speaking?
The voltage channel gates.
Exactly. So what's going to happen? But remember, what causes them to close?
Repolarization.
Excellent. With no new neural action potentials, the neuron, and in particular the synaptic end bulb, repolarizes. And then you guys are absolutely correct. When the synaptic end bulb repolarizes, our voltage gated calcium channels close, or let's even be more specific, drop below threshold and close. So that means there is no new calcium into the synaptic end bulb. We got no new calcium coming in, but we still got a lot of calcium in there. So what do we have to do with that?
Potassium pump, the calcium pump.
Calcium pumps remove calcium from the synaptic end bulb. And when we remove calcium from the synaptic end bulb, what's the effect of that?
No exocytosis.
No more exocytosis of acetylcholine. Excellent. From the synaptic end bulb. So there is no new ACH into the synaptic cleft. Excellent. What's next? We're not putting any more acetylcholine into the synaptic cleft, but that doesn't totally resolve our issue. What do we need to do?
The enzyme.
Sure, what does the enzyme do?
The break the acetylcholine, which is acetylcholine esterase.
Excellent. So you got the right idea. So there's no new acetylcholine going into synaptic cleft, but we must remove the current acetylcholine that is there from the synaptic cleft. And you are correct. One of the main ways we're going to do that is with that enzyme acetylcholine esterase. Breaks it down. However, are we reliant just on acetylcholine esterase to get rid of our acetylcholine?
No.
Now, how many different ways do we have?
3.
3. Excellent. So there are 3 ways. So notice, if something happened to our acetylcholine esterase, we would still be able to get rid of acetylcholine. It would just be a lot harder. It would take a lot longer. But we do have 2 other ways to get rid of it. What are the 2 other ways we can get rid of it?
Endocytosis.
Fusion.
Excellent. Excellent. Perfect. You guys got them both. Endocytosis back into the synaptic end bulb, and diffusion away from the motor end plate. Because once it's away from the motor end plate, it can't affect the muscle cell anymore. So now, thanks to that, there's no acetylcholine in the synaptic cleft. What's the immediate effect of no more acetylcholine in the synaptic cleft?
chemically gated channels close in the map, motor end plate.
Excellent. The chemically gated cation channels on the motor end plate close. Perfect. I may need some more room, so let's do that. Excellent. What happens when they close? Immediate effect of closing those chemically gated cation channels.
Repolarizes.
Why does it repolarize? Something's got to happen first before it's able
Sodium.
to repolarize.
The sodium.
Yeah, so if those channels are closed, can sodium enter the motor end plate anymore? No. So no new sodium enters the motor end plate. Remember also, no new potassium can leave either. But was it the leaving of the potassium that really affected the change in the muscle cell? No, it was all that sodium coming in. And so then you're right, using pumps, etc., the motor end plate repolarizes. And when the motor end plate repolarizes, drops below motor end plate, drops below threshold. And when it drops below threshold, no new muscle action potential is produced. What's next?
No more.
No contraction. No new action potential, or we can say.
Yep, so we have no new, new, muscle action potential is produced at the motor end plate. So
It doesn't travel down the sarcolemma to start the next step or phase.
excellent. No muscle action potential down the sarcolemma or down the transverse tubule. And if there's no muscle action potential driving down the transverse tubule, what happens next?
So the gate is the no depolarization.
Right, exactly. So the triad is no longer depolarized, and when it's no longer depolarized, what happens? Goes back to rest, and what was the fancy term for that again?
Repolarize.
Yeah. Triad repolarizes. Of course, we should probably say what that triad is. The triad is the T-tubule plus the two terminal cisternae. So the T-tubule plus the two terminal cisternae repolarize. And when the triad repolarizes, you are correct, our voltage gated calcium channels on the sarcoplasmic reticulum close. That means no new calcium into the cytosol. What happens next?
Calcium can't bind to the troponin to move them and shape them.
You are correct. That is going to happen. But remember, while there's no new calcium entering in the cytosol, there's still calcium in the cytosol. So to get the calcium to stop binding that troponin, we have to get rid of that calcium first. So how do we get rid of the calcium in the cytosol? Pumps.
Gets pumped
Calcium.
back into the sarcoplasmic reticulum.
Excellent. Calcium pumps return the calcium into the sarcoplasmic reticulum. And I can abbreviate because I've already written it out once. Into the sarcoplasmic reticulum. And then you're correct. With no new calcium in the, or with no calcium, sorry, the cytosol, then what happens? we don't have calcium in the cytosol anymore, what was that calcium doing?
Opening the passes calcium.
Well, remember when calcium was in the cytosol, what was it doing in the cytosol?
Binding to the troponin for the conformational change.
Right, changing the shape of the troponin by binding to the troponin with no calcium. So with no calcium in the cytosol, our troponin is going to go back to its original shape. And when it goes back to its original shape, our troponin is going to rotate that tropomyosin back over the, and let's for brief, for space, active site on the actin. And if it's binding to, if the tropomyosin is covering the active site of our, on our actin, then can myosin grab the actin anymore?
No.
No. Primed myosin heads can no longer form a cross bridge with the actin. And if it can no longer form a cross bridge with the actin, no more tension, no more power strokes. And if there's no more power strokes, no more tension, and our muscle relaxes. And there you go. Just that easily, we have described the process step by step of how our muscle cell is going to relax.
We have this information, like a copy of this information in the canvas.
No, because again, I don't want you to memorize the words that I use to write this. This is a process. It really, if you think about it, there's a lot of material on here, but it's no new information. As we mentioned, this is basically steps 2 and 5 together. In fact, let's do that, right? To remind ourselves of this, if we were to terminate, let's make this smaller. Termination of communication at the neuromuscular junction. Oops. Where would that start in this process that we have written out?
Making the decision.
Yeah, so this is the start of that. And where would it end?
The muscle action potential being made.
Excellent. So let's cheat and do this, that, and that. There we go. No new muscle action potential is produced. So this would be the stop of termination at communication of the neuromuscular junction. But not only is this the stop of the termination of communication at the neuromuscular junction, but if we were to talk about, oops, no, hold on. There we go. If we were to talk about termination of excitation contraction coupling, where does that start?
So from binding troponin with the actin sites.
That's where it ends, but where does the termination of excitation contraction coupling start?
The termination, okay.
If we're terminating excitation contraction coupling, where would that process, if I asked you that for the essay question, where would the first thing you write be?
It would start at where the MEP drops below threshold.
Exactly. Remember this, because it is a continuous process. When we're describing this processes, what is the end of one step is the beginning of the next step. So this would be the start of termination at excitation contraction coupling. And where would it end?
Troponin, troponin rotate, tropomyosin.
There you go. Blocking the binding site. So notice these are just the two processes, 2 and 5 put together. So there really isn't any new information here. It's just this is also a reminder that this is indeed a continuous process. We've broken it up into more smaller, easily digestible pieces. But it really is one continuous process. And we see this when we write it out that way. All right. Questions on that? All right, excellent. And then just that easily, we are done with all of our processes. So as I mentioned, our goal now is to switch gears and talk back about the muscles again. And for these muscles, again, we started with the muscles that move the head and the neck. We are now going to talk about muscles that affect the core of the body. All right, starting first with the trapezius. Notice the trapezius is a large, superficial, mostly posterior muscle. Let's write that out. Large, superior, superficial, and mostly posterior muscle. And there's that pesky word in there, mostly, because what does mostly mean?
Not all the time.
Not all of it, not all of it, absolutely. And again, I think we get a nice job of seeing this when we look at its origin and its insertion. We'll identify those in a minute, but notice we have this big converging muscle. Highlighter. So notice again, we have, here is our origin, and then we have all these fascicles coming together from all these different orientations, which not surprisingly gives us a wide range of motion. So not surprisingly at all, it is going to have, how many actions does this muscle have? 4, right? So there's a whole lot of stuff going on in this big convergent muscle. But let's look at its origin and insertion, because as I mentioned, the origin and insertion helps us to understand the actions. So let's switch to this view. And our trapezius is located here. Perfect. So I want that, and I want that. Oops, hold on. Perfect. Excellent. So what is the origin of the trapezius?
The occipital bone and the C7 and all the thoracic vertebrae.
Okay, excellent. So it starts up here on the occipital bone, right? That external occipital protuberance. Excellent. What else?
C7 and the thoracic vertebrae.
Excellent. So the spinous processes, this posterior process of C7, T1, T2, T3, T4, T5, T6, T7, T8, T9, all of them?
Yes.
There you go, 10, 11, and 12. Excellent. Oh, it looks like I missed one there somehow. All right, but you get the idea. So again, you can write it out. You're not going to be drawing them, so just write out C7 through T12. Anything else? Excellent. Ligamentum nocha. Notice it's not connected directly to the cervical vertebrae except for C7 with that nice big vertebral prominence, the spinous process that sticks out and was one of the bone features you were responsible for on your 30-point review. And that makes sense. We don't necessarily want these big, huge muscles connected directly to the bones of the neck because that would limit our range of motion. Luckily, we have that nice big elastic ligament, though, that sits over top of these muscles. Over these bones. So it doesn't connect directly to the cervical bones, except for C7, but it does attach to that elastic ligament over the top. So notice because of that, it basically has this big, long, straight origin from the top of the head all the way to the back of the thoracic vertebrae, right on the midline. In fact, that's kind of where it gets its name. If you notice when you look at it, let's cheat and go back to the, if I leave, this picture's gonna go away. It's a big trapezoid shape muscle. In fact, when you put them both together, you have this big, huge kite on the back of the body up here, superior and posterior. So that is our origin. What is the insertion?
Continuous insertion along acromion, spine of scapula, and lateral third of clavicle.
Excellent. And again, that phrase continuous insertion isn't something you would necessarily have to write on the exam. It's more of a reminder to you that, again, by continuous insertion, it means it goes all the way along. So it goes along the spine out to the acromion. And then notice it actually comes onto the anterior side of the body, onto that lateral third of the clavicle. So notice just the littlest bit of its insertion actually comes onto the anterior part of the body. So that's what we mean when we say it is mostly posterior, because a little bit of it does come onto the clavicle on the lateral side out here. All right. Now, we've drawn the origin and we've drawn the insertion. We know this is a convergent muscle, so we're going to have all these fascicles that are going to connect from the origin to the insertion. And notice, what part of the body does this influence and move? Your shoulder, your scapula. Notice it doesn't connect to the humerus. So it's not swinging the arm around anywhere in space. Almost died. Swinging your arm around too much in space. It's about moving your scapula, moving your shoulder. And that is why this is considered a trunk muscle. It is not connecting to the arm directly. It is instead connecting to the shoulder. Now, we've drawn the origins and insertions. Let's go back and look at the pictures because there's a couple things I want you to see. Notice as you look closely at this illustration, you can see how some of those fascicles are going and traveling towards the front of the body. We can see it if we go back to that chart. Notice as we look at the chart view, we can actually see some of the trapezius actually sneaking forward onto the front of the body here. But again, notice with the picture from your textbook, with the illustration, the chart from the classroom, with the picture from the textbook, we see subtle examples of this. But what if we get one of those big, huge behemoths that we've talked about? Notice with these bodybuilders whose job it is to dramatically enhance these muscles, we can much more clearly see that anterior portion of the trapezius that is coming over the top of the shoulder onto the anterior side. So it makes it much, much easier to see on this bodybuilder. It's a big, obvious muscle coming across onto the anterior side with that shoulder. All right. We talked about its location, its origins, and its insertions. Any questions on that? All right. We heard that there are 4 actions. What are the 4 actions of this muscle?
Raise, rotate, retract the scapula, and then extend the head.
Excellent. And notice I have it written that way. Notice those aren't exactly the appropriate anatomical terms. So let's use the appropriate anatomical terms first. elevate the scapula. Right, so it's going to elevate the scapula, bring the scapula up. It is going to, and that's the one that we used as raise, right? Elevate the scapula. So that's the raise one. We are going to retract the scapula, pull the scapula back towards away from the front. And remember, as we do that, we also bring it towards the midline. So wasn't there another term we could use that was equally appropriate to retract when talking about movement of the scapula? You know, if we're bringing it towards the midline, remember that's going to be, if we're bringing it back.
Abduct.
Exactly, a deduct the scapula. And the third one was to rotate. You can kind of feel this for yourself, but it's actually easier to feel it on somebody else. So what I recommend you do is find someone in your living space to put your hand on their shoulder blade. If you can't do that with them, then go find some random person on the street. Make sure you're both wearing your masks, but ask them if you can palpate their scapula. And as you put your hand on the back and as they abduct their arm, what you'll notice, if you think of the skeletal system, and let's cheat and actually look at the skeletal system for this. This is actually a good example. So let's look here. Notice as we were to deduct the humerus, as we were to bring the humerus up, notice eventually the head and neck of the humerus would reach a point like that. And if you continued to try to move it up, you would smash the greater tubercle and the neck into the acromion and into the clavicle. And do we want to do that? No. Does that mean you can only abduct your arm to here and you can't bring it any higher? No, of course you can abduct it higher than that. But to bring it higher than that, what we actually have to do is we actually have to rotate the scapula. So what'll happen is as you continue to bring your arm up, eventually you have to rotate the scapula. And so that's what you feel. If you put your hand on someone's shoulder blade, as they slowly abduct your arm, you'll see at first just the arm moves, but then the scapula starts to rotate so that you can bring the arm all the way up. So those three key actions are to elevate, bring it up, retract, bring it back, or abduct it, and rotate. Now, again, I know raise isn't one of the appropriate terms, but I use it because raise, retract, and rotate are commonly thought of as the 3 R's of the scapula. All right. Back in ancient times, and in ancient times, I mean when I was in elementary school, we learned the 3 R's in elementary school. And what were the 3 R's that we learned in elementary school when I was little?
Reading, writing, arithmetic.
There you go. Reading, writing, and arithmetic. And if you think about it, right, 2 of those 3 things aren't really R's. Right? Writing and arithmetic, neither of those things are R's. So it tells you something about the education we get. Allison, I appreciate you thinking I'm that young, but that's more what my daughters have been learning, right? My daughters now learn about recycle, reduce, and reuse. Exactly. So that's their 3 R's. Well, your 3 R's are raise, retract, and rotate. Those are 3 of the 4 actions of our trapezius. -2 participation points for Allison. All right, excellent. What's our 4th action of the, that is the best joke so far in class. Excellent. So what is the 4th action of the trapezius? No, look, that's hilarious. I agree with Haley. Best joke of the class so far. What's the 4th action? Extend the head. Notice if we were to just use the fascicles from the posterior and superior part, if you think about these fascicles that are connected here, if we just contract those, then what we're able to do is we're able to extend our head. And so if instead, remember, as we talked about, normally we pull the insertion towards the origin. But as we've talked about, if we stabilize our scapula using other muscles and just contract the superior fascicles of the trapezius, we can bring the head back, we can extend the head. So those are the 4 actions of the trapezius. I know hopefully you guys are working on that handout that I have. Has anybody come up with an activity where you might do all 4 of these actions together for the trapezius, a common or uncommon activity that can help us all to remember what our trapezius does for us? Maybe I'll start giving out extra credit next week for people who volunteer to demonstrate some of these activities for us. Okay, it may not, oh, fair enough, it's not on the list, but it's still a good way to cheat and learn this material, good mnemonics to help you. So can anybody think of one off the top of their head? Well, I'll give you the one that I think of. Like I said, maybe next week I will encourage people with extra credit for who come and demonstrate the activities. So the one that I always think of for this one is a serve in tennis. Notice if you're getting ready to serve the ball in tennis, what you have to do is first get the racket into position. To do that, you have to elevate your shoulder. You have to bring your shoulder back, and you have to rotate the scapula so that your racket is up there ready to hit it. Of course, if you're going to make that serve after you bring it back like this, when you throw the ball up into the air, what do you need to do? You need to look up to see it. So when you look up to see it, you are using that trapezius. You're right, Haley, getting ready to pitch. When you're getting ready to throw that ball, you're throwing out that first pitch at the game, absolutely. You're getting it back. So it's not the bringing forward action, but the bringing it back, getting ready to pitch, absolutely. So whether you're getting ready to throw that pitch or whether you're ready to hit that tennis ball, you are using that trapezius to get that arm back there and get it ready. Yeah, volleyball works too. Any of those things that helps, absolutely. The only thing I don't like about the pitch is usually when you're pitching the ball, you don't look up when you're doing it, right? So that's the nice thing about a volleyball. The nice thing about a tennis ball is you have to look up to see the ball to make that contact. So I like that. Volleyball, tennis ball, something like that. All of those work.
So what,
Yeah. When you're throwing the pitch, you want to make sure it's a strike. Yeah, you're praying for a miracle. Look up. There you go. Exactly.
come up with what common word or activity for this?
So again, there is no right answer to this. These are just tricks to help you to remember these things on the exam. If on the exam, I ask you for the 4 actions of the trapezius and you write serve a volleyball, are you getting any points for that on the exam? No. So it's just a trick to help you to be able to remember that. If your example wants to be beating your kids, right? You get in your hand up there to beat your kids, whatever makes you happy. Use whatever you like. I don't care. The trick is for you to come up with something that helps you to be able to know and remember what these things are. Okay, so I don't care what your examples are. These are just tricks for you to help you to be successful, to remember this material. Because we have so much material, mnemonics. Again, some people are good at just brute force memorization. And if you work that way, congratulations. I don't. I like tricks. I like associations. I like mnemonics because they help me to retain information. I'm a very visual learner. I'm a very tactile learner. So if I can put these things into my hands, if I can think of physical activities associated, or I can visualize these things in my head, it helps me to remember this thing. So again, it's just a different way of learning this material. And so it's whatever helps you to be successful. Okay. And it breaks up the boringness of everything else. And I get my exercise button for the day by swinging my arms around all over the place as we talk about these things. All right. That is the trapezius. Any questions on that one? All right, let's look at the next muscle on our list. The next muscle on our list is the serratus anterior. Now, before we draw this one, I want to actually draw the origins and insertions. I want to make a separate drawing for this one as well. So let's steal the whiteboard back. Because I think the organization of this muscle, I think is one of the trickier ones we have to deal with. So here is my rib cage. And just for simplicity, we will put the vertebrae back there so that we know that this is posterior. And then the anterior is up here. And as we've already talked about, we know that the scapulus, those shoulder blades, are located here, not attached to the ribs, but associated with the ribs on the backside. Now, what is the origin of the serratus anterior? Excellent. The anterior surface. So what we can do is we can kind of draw here on the anterior surface of one of the ribs that we're looking at. But you're right, all the way 1 through 8, and we will draw those on the actual picture. But I want to show this to you this way first. What is the insertion of the serratus anterior? Again, you have these lists right in front of you. This is your opportunity to merely read something off a list, participate in the class, and have everybody be impressed by how smart you are. Excellent. Anterior surface of the medial border of the scapula. So let's think about this. This is the scapula. This is the anterior side. And remember, over here is the medial border. So here on that medial border of the scapula, underneath the scapula, near the ribs, is where the insertion is. So if you think of the muscle, what this muscle does is it wraps around the ribs. It doesn't attach to the ribs. It wraps around the ribs and attaches to the posterior side of the scapula. Now, when we contract muscles, we know, of course, that we bring the insertion towards the origin. So, of course, what type of action would that be? We'd be bringing the scapula forward, so that would be a protraction. Excellent. However, notice, is it really going to be able to pull its way through the ribs and come in this way? No, of course not. So notice what it really does is kind of pull it out towards the side. And so when it kind of pulls it away from the midline, what is that other term we could use for this? We could say that it protracts the scapula, or we could also equally say that it
Abduct.
abducts. Excellent. And again, I emphasize the D and the B to emphasize the difference. So it abducts the scapula. Excellent. So it can pull it forward, but the other thing that it is going to do is it is going to put tension on the scapula. So the second action is to hold the scapula against the ribs. Hold it against the wall of the thoracic cavity. The nerve that controls this muscle passes through your neck. And what can happen is if your arm gets pulled or twisted, or you get a compression from falling on it, you can damage that nerve. And what happens is if you can't put tension on the serratus anterior anymore, what'll actually happen is that the scapula will actually rotate and point out backwards. It'll actually come off of the ribs, come off the thoracic cavity. It's a condition we call a winging of the scapula. And if your scapula wings out, are you going to be able to freely move your shoulder as easily as you could before? No. So we have these two actions. One is the movement of it, bringing it forward or bringing it this way. And the other is to hold it in place against the wall of the thoracic cavity, stabilizing your scapula, which helps to stabilize your shoulder. All right. This is what it looks like from the top. Of course, we need to draw it on our pretty picture. So if we come here and go back here to where the serratus anterior is, as you guys have already told me, the origin is on the anterior surfaces of ribs 1, 2, 3, 4, 5, 6, 7, and 8. And this is actually where it gets its name. If you look at it, if you think about it, all the individual muscle heads that come in and attach give this kind of a scalloped or serrated edge, like a serrated knife. It has this serrated knife appearance to it. And that's where it gets its name. Serratus anterior is the serrated looking muscle located on the anterior part of the ribs. Now, its insertion we know is the medial border. Technically, we can't see the medial border. So again, you draw what makes sense to you. If you want to use a lighter color to remind you, or a dotted line to remind you that it's going to be back there, that would be a fine way to do it. If instead you wanted to just draw a dark line just at the edge where you could see it, again, or if you just wanted to draw that circle picture that I just drew, where we drew a circle and you drew a scapula, and then you put it on the edge of the scapula there so that you remembered. However you want to do this, again, you're not turning these in for a grade. These are your study guides to help you to be successful on this exam. So draw what makes sense to you. But we know it is on that anterior medial border of the scapula. All right. Now, one last thing. Notice, to see the entire serratus anterior, we have to cut and remove many of these superficial muscles. This is a deep muscle, mostly deep muscle. And so, of course, what's the key word with something being mostly deep? What does mostly mean? Not all. And so notice over here on the right-hand side, with all those superficial muscles intact, notice we can still see a couple of the bellies sticking out here underneath. This is going to be especially true if someone elevates their arm, or really, extends or flexes their arm all the way up. Notice, for instance, on our, okay, here again, we can see it deeply with it being removed. And what I like about this picture is by removing all the other muscles, notice they can show us where it is actually inserting into that medial border of the scapula out there. So that's been nice that they removed that for us. But notice in our, we'll come back to that. Here, with our arm flexed, notice on our chart, we can see several of the bellies of the serratus anterior actually sticking out. But could you really see something like that on an actual person? Yep. Notice, while it is a mostly deep muscle, we can actually see some of the inferior bellies of the serratus anterior. Okay, well, maybe he's not a normal person. That's pretty much my physique, but again, most, maybe some people, but yes, in some individuals, maybe not on a normal person, but on some individuals, we are actually able to see that serratus anterior, those inferior bellies of the serratus anterior. Oh, absolutely. Yeah, in fact, I have some good female pictures on this that we'll take a look at in a little bit, but actually not till we get to the arm. But yeah, sure, absolutely. All you have to do is grab one of those bodybuilder magazines, or heck, the websites and stuff like that now. Do a web search, woman bodybuilders. I'm sure you'll find plenty of interesting pictures if you do that. Make sure your child safety controls are on when you do that. All right, excellent. Questions on this muscle? Notice, remember, one of the things that I mentioned is that you are going to be responsible for identifying muscles on a bodybuilder picture, like this. Technically, the serratus anterior is a deep muscle. But even though it's technically a deep muscle, it's really a mostly deep muscle. So is this a muscle that, even though it's technically deep, I could ask you to identify on the exam? Yeah, absolutely. So again, while most of the muscles you're going to be responsible for are superficial, there will be a couple tricky deep ones like this that you would be responsible for as well. Excellent. So here we get this great superficial view of the trapezius. And now deep to the trapezius, and so here it's been removed on this side, we see our next muscle group. This muscle group is made up of two muscles, and those two muscles are the rhomboid minor and the rhomboid major. And do we care about those names of the individual muscles? No, we just need to know them collectively as the rhomboids. Even though this does a nice job of showing it, let's go ahead and draw our origins and our insertions. So what is the origin of the rhomboids?
Spinous process of C7 through T5.
So again, here on the spinous process of 7, T1, T2, T3, T4, and T5. Excellent. What is the insertion?
The posterior medial border of the scapula.
There, excellent. Once again, we're on the medial border of the scapula, but this time on the posterior side. So that, notice, our fascicles get pulled towards the midline. Or notice, again, hopefully you've got that picture, you can do that, but let's go back to this picture. Remember this picture, we just need to add one more thing to it. We need to add our spinous process of our vertebrae here. And if we do that, we can actually see where the rhomboids would be located. The rhomboids are going to originate from the spinous process of our vertebrae. Move this over here. And it is going to insert into the posterior medial border. And so this muscle basically, oops, don't want to use that. Let's use green. Connects here. And notice this basically is going to pull the medial border towards the midline. And what kind of action would that be? A deduction. But notice as it's adducting it, it is also going to pull the scapula back. And so, of course, that would be a retraction. And so notice that is what our rhomboids do. Our rhomboids bring the scapula back. These two, notice, both help with the scapula. The way I always think of them is, like I said, I think we used this example already, but I always like it, right? Pretty soon, eventually, the next year or so, we're all going to go out and see each other again. And when we do that, we're going to be super happy. You're going to see that best friend you haven't seen in a year, and you're going to go give him that big hug. And as you're giving him that big hug, you are using your serratus anterior to bring your scapulus forward, right? If you hug all the way across, you can feel your scapulus coming out. Of course, then you realize you haven't seen them in 9 months because of the coronavirus, but then you remembered that just before you guys went out on this big expensive dinner and they owe you $200. So what do? You use your rhomboids to bring your arm back so you can punch them in the face, right? So again, the rhomboids bring the scapula back, the serratus anterior bring it forward. So these two both attach to that medial border and pull the scapula back and forth. Absolutely. So this is exactly, this is the grandmother muscle. Absolutely. When she tells you not to slouch and tells you to bring your shoulders back, that is this one. Absolutely. This is the grandmother one. And again, this is also when you're rowing, when you're doing those rowing exercises, as you bring it back with both arms, you can feel these muscles bunching together in your in the midline of your back as you're pulling both scapulas back at the same time. Absolutely. All right. Questions on that? Excellent. Let's move to the next one. Pectoralis minor. Our pectoralis minor is a small muscle. And again, anytime we have a minor, what do we also have? A major. We have a pectoralis major. But I don't see the pectoralis major on this list. Do you? Well, not on this page, but maybe on the next page.
The upper extremity one.
Yeah, the pectoralis major moves the arm, whereas the pectoralis minor moves the shoulder. And that's why the pectoralis minor is on the trunk list where the shoulder is not. The pectoralis major is not. Again, we see this really nicely when we look at the origins and the insertions of it. We can see how while they may be similar in name, they're very different in their function. What is the origin of the pectoralis minor? Again, list in front of you. These should be the easy questions. Absolutely. Anterior surfaces of ribs, 3, 4, and 5. Excellent. What is its insertion? Excellent. That bird's beak coracoid process of the scapula. Notice it doesn't attach to the humerus. It doesn't attach to anywhere on the arm. It attaches to the scapula. It moves the shoulder. And as we know, we bring the insertion towards the origins. So when we contract this, I got to bring my camera down, it is going to bring the shoulder down. And what's the fancy word for bringing the shoulder down? Excellent. So it is going to depress the scapula. And because of where it's located, it can also bring the scapula forward. What's that name for bringing it forward? Protract the scapula. And again, it completely interchangeably with protract. What else could we have said as well? When we bring it forward, we also bring it away from the body. So it would also be in abduction. So either of those are acceptable. So it brings the shoulder down, it brings the shoulder forward. However, remember, as we also talked about, we can reverse the actions of the muscles by stabilizing the shoulder. If we hold the shoulder in place, like with my trapezius or other muscles, then when I contract this muscle, what'll happen is it will bring the ribs up towards the coracoid process, and it elevates the ribs. And when you elevate the ribs, obviously that allows you to inhale. So it's, again, one of those accessory muscles that help us to inhale. This one can be a little tricky because there's a lot of stuff involved in it. Can anybody think of an activity where you would use this muscle in those 3 actions? You definitely want to stabilize yourself when you bench press, but getting ready to bench press probably, right? But again, you got to be careful because the bench press is this action. So you're right. I think locking in your core to get ready to do a bench press, I think that that's a pretty good example. I like that one because you're kind of bringing the shoulders down, you're bringing them forward, you're inhaling, you're getting ready. So you're engaging your core so that you can do that bench press. So it wouldn't be the bench press itself, but preparing for the bench press. I think that's a great one. That's actually one of the better ones I've ever heard. This one's challenging for me. I'm confused by a little bit. My sister tells me she uses this muscle to get out of tickets all the time. But I got pulled over by a policeman the other day and I depressed my scapulus, I protracted my scapulus, and I elevated my rib cage and I still got the ticket. So I'm not sure why it works for her and not for me, but I like the bench press example a lot. So maybe that's a better example. All right, questions on that? All right, excellent. Perfect. So that is that muscle. And notice, just that easily, we have done all the muscles that we need to know the origins and the insertions for of the trunk. So questions on any of that? All right, excellent. How are we doing on time? We're doing pretty good.
So only for muscles, right, sir?
I'm sorry?
Only 4 muscles in the trunk.
Well, that we need to know origins and insertions for. I think that was all of them. Let's double check. Yep, looks like there's only 4 that we need to know the origins and insertions for. Notice we're certainly not done with all of the muscles of the trunk, but we are done with all the ones. So there's only 4 you need to know the origins and insertions for on the core of the body, on the trunk of the body. All right. But notice we have a lot of other muscles here on the list that we need to talk about. So let's talk about some of those, starting first with the diaphragm. We talked about the diaphragm on the very first day of class. We talked about how it is that anatomical barrier between the thoracic cavity and the abdominal pelvic cavity. Notice when we look at this picture, we see how it forms that boundary, but notice also there are some structures that do need to pass through it. Notice there's a small opening for the blood vessels, the inferior vena cava and the aorta. There's also an opening for some of the nerves to pass through and also our esophagus, because after all, our food has to get through the thoracic cavity into our stomach. But for the most point, it is this big, huge physical barrier that separates the thoracic cavity from the abdominal pelvic cavity. And it is a bell-shaped or curved shaped muscle. So notice, for instance, if we were to go back to the whiteboard and clear this and draw a box. Here is our abdominal pelvic cavity. And we have this physical barrier, this bell-shaped curved muscle. That's horrible. Let's see if I can make it a little bit better. There we go. This bell-shaped curved muscle that separates the thoracic cavity from the abdominal pelvic cavity. Now, it is a curved muscle. So what happens to all muscles when they contract?
Shorten.
Yeah, when a muscle contracts, that muscle gets shorter. When you have a curved muscle and it contracts and gets shorter, what happens to it?
Flattens.
Yeah, it's going to flatten out, exactly. So when this muscle contracts, it is going to cause the muscle to flatten out. Notice this is a contraction. So that means this is an active process. What does it mean to be an active process again?
ATP.
Yeah, it uses ATP. Excellent. So it uses ATP. Notice when it flattens out, what that is going to do is it is going to increase the volume of the thoracic cavity. Why is that significant?
Deep breath.
Pressure.
Well, pressure. When you change the volume, you change the pressure. Absolutely. So if volume goes up, what happens to the pressure?
Goes down.
Yep. And remember, pressure is what makes the world go round. Again, we have to assume we're in the classroom. Assume we're in the classroom. When we talk about pressure, especially when we're talking about gases, if all 30 of us or 21 of us that are here right now were in a room together, and what I would do is I would blindfold you guys and I would let you wander around the room. And every once in a while, you guys would bump into the wall. And I would count the number of times somebody bumped into the wall. And that basically would be the measure of pressure that we had in that classroom. If instead I took all of you and brought you here into my bedroom is a lot smaller than the classroom. And so what would happen to the number of times you bumped into the wall?
Many.
It would go up, pressure would go up. If instead I blindfolded you and let you roam around Sacramento, how often would you bump into the boundaries of Sacramento?
Less.
Not very much. So as we change the volume, the pressure changes. So in this case, with the decrease in pressure allows, because, oh, I guess I have to draw it, right? This is connected to our mouth on the outside, which, and of course, you can see that is the mouth. And of course, when that pressure decreases in the thoracic cavity, air is going to come in. And of course, this process we call an inhalation. All right, we inhale. All right. So this is that motion, the active function of our diaphragm is to increase the volume of our thoracic cavity, which decreases the pressure, which causes air to move in. We get that inhalation. And like I said, while you're sitting here in class, in your chair, listening to the lecture, like I said, unless you're doing this on a treadmill or using one of those standing desks, as you look down, you don't see your chest heaving while you're taking your normal resting breath, because this is half of your normal resting breath. Of course, the other half of your normal resting breath is when we relax the diaphragm.
It means.
Go ahead.
So when the muscle has the function or job of inhalation, does it function also? It functions like for the exhalation.
Well, that's what we're going to do right now.
Okay.
So we contracted the muscle, and when we contracted the muscle, it came flat. But now we're going to relax the muscle. When we relax the muscle, relaxation of the diaphragm, because muscles are elastic, it's going to go back to its original shape. And so what's going to happen is it is going to go back into being a curved shape muscle. So it is going to go back to its original shape. So the muscle goes back to original shape. Now, is this an active process?
No.
No. Remember, this is a passive process. This is just elasticity of the muscle that is allowing this to occur. So there's no ATP that is used in this process. However, when we relax that muscle and it goes back to its original shape, that causes a decrease in the volume of the thoracic cavity. And that, of course, causes an increase in pressure in the thoracic cavity. And because this is, again, connected to the mouth, when the pressure changes in here, does that air want to stay in here anymore? Nope.
No.
And so that air is going to leave. And of course, that is the process of exhalation. So notice the diaphragm technically is responsible for both breathing in and breathing out, what we actually call your resting or tidal breathing. However, is it actively involved in both?
No.
No. So technically, it's active action where it's using ATP, which is what we care about what a muscle does, is that it increases the volume and causes inhalation. And so while you're sitting here at rest, you are using these muscle as your primary muscle for your breathing process. And notice we only use ATP for half of this. Notice one other thing. This works because of the elasticity of the diaphragm. The diaphragm goes back to its original shape. The ribs go back to their original shape. The lungs have elasticity and go back to its original shape, which is great because when you stretch out a rubber band, you let it go and it goes back to its original shape. But what if you stretch out that rubber band 10,000 times? What starts to happen to its elasticity?
Losing elasticity.
As we age, we lose elasticity, right? And then there's the 20 years that Grandpa spent working in the coal mine. So he was inhaling all those that coal dust, damaging his lungs, causing scar tissue, right? The injury from that fall in that happened. One of the things that happen as we age is that the thoracic cavity loses its elasticity. Now, luckily, we have accessory muscles that can allow us to take a more exaggerated or a more purposeful exhalation. But the problem becomes is now for Grandpa both to inhale and to exhale, he has to use ATP. He's basically using twice as much ATP to breathe as he was using before. So one of the things you may notice about Grandpa is just sitting in the chair breathing, Grandpa's exhausted. Why is he exhausted? Because he's using a lot more energy than you are just for the simple act of breathing because of that loss of elasticity. All right. But again, you don't have to be Grandpa to take an exaggerated breath. As we've talked about, we have extra muscles that can help us to take an exaggerated inhalation or extra muscles to make a more forceful exhalation. We've already talked about some of these. What are some of the accessory muscles that we have already identified so far in this class? I can think of at least 3.
Scalenes?
Yeah, scalenes. Aging does limit our ability to maintain homeostasis, and that is one of the things that ultimately leads to the termination of our life. I think that's more of a deep philosophical question that you've asked. And again, why is always one of those questions that's hard to answer in anatomy and physiology, because the engineer who designed all of this didn't leave us her footnotes. So why is something we can try to infer and interpret, but we're not always going to be able to answer. Excellent. Scalenes was one of them. I can think of at least 2 more.
Sternocleidomastoid.
Excellent. And one we talked about today.
The pectoralis minor.
There you go. I will warn you that there are others we haven't talked about yet, but this is a good starting point. It reminds us of things we've talked about. We didn't emphasize it at the time, but there is an exhalation accessory muscle that we briefly mentioned and didn't kind of emphasize the importance of it. And that muscle that we were talking about earlier was the Serratus Anterior. Notice as we talked about, it squeezes the scapula against the walls of the thoracic cavity. And as you're squeezing the scapula against the walls of the thoracic cavity, that helps to squeeze the thoracic cavity and can be used to force air out as well. So these are not the only accessory muscles, but these are the ones we've talked about so far. Let's add some more to it. All right. So, questions on the diaphragm before we move forward? I'm making that mistake that they always tell lawyers not to do. Don't ask a question you don't know the answer to, but I'm going to cheat and let's take a look because I'm curious about something on that website from Cosumnes River. I'm hoping it has it because I think they do a pretty good job of having some torso stuff here. So here again, torso model of the muscular system. Excellent. So they got this part. This is our torso model. I pick on our model makers and our artists a lot because they don't do things as well as they could. This one is really nice. We can see the pectoralis minor. We can see the serratus anterior. I'm hoping, oh, here we go. There's the serratus anterior really nicely. Pectoralis minor really nicely. But what I'm hoping they have, they don't have the inside view. Shucks. Oh, well, okay. There's a nice view of the diaphragm. So we see that really nicely. And then we can also see on the posterior wall, we can actually, no, actually it doesn't show. Yeah, it does show a little bit. They don't have it labeled, but up here is where the diaphragm would come off. On these models on the inside, I wonder if I have that. I've given you some pictures on our website. Let's check to see what we have there. I don't remember what I've put there. Oops. Yeah, okay, I don't have it. But notice again, as we talked about, you do have the leg, some of the leg models that we'll talk about. We do have some of the classroom models. So we do have some things there that we can look at. They don't have the inside of that abdominal, but if you look at the inside of that abdominal model, you can actually see the curved muscle of the diaphragm where it connects to it. So I'll see if I can find a picture for that for the next class that we can look at for that. All right. Any other questions on the diaphragm before we move to the next one? So we've seen the muscle, we know where it's located, and we know its function. Excellent. So let's talk about some of the other accessory muscles. Two of the other accessory muscles that help in breathing are the external intercostals and the internal intercostals. Now, I like this picture from your textbook, and your textbook's got an even better one, but I also like simple pictures for a starting point. So let's get a simple starting point picture. That simple starting point picture we'll draw ourselves. So of course, it needs to start with the sternum located in the middle. And then of course, we need ribs. And even though I know the abdominal pelvic cavity is curved, the thoracic cavity is curved, I am going to draw my ribs straight because it is convenient for me. Now, as we know, these ribs are connected to the sternum directly. These are ribs like 2 and 3 or 3 or 4 or something like that by hyaline cartilage. So we know hyaline cartilage connects these together. So this is a very simple, very basic diaphragm of what the rib cage looks like. And make a point of emphasizing this because there are two muscles we need to talk about. We need to talk about the external intercostals, and we need to talk about the internal intercostals. For the external intercostals, there are 3 main characteristics we can use to identify them. As the name external would indicate, they are superficial, closer to the surface, which is why they're external. But they are also more lateral. So these are superficial and they are lateral. The third characteristic we can use to identify them is the orientation of their fascicles. The fascicles of our external intercostals, let's actually do this on a separate line. The fascicles of our external intercostals project down and in. So if I were to draw my external intercostals, they're going to be superficial, they're going to be lateral, and their fascicles project down and in. So we have this down and in projection. And notice, because they're lateral, they are not going to come in contact with the cartilage. Same thing here. We're going to go down and in with their orientation between the ribs. We don't have to worry about origins and insertions, but they're located between the ribs, as the name internal intercostal would indicate. Conversely, the internal intercostals are going to be deep because they're more internal, and they are going to be more medial. And their fascicles project down and out. Oops, I spelled project correctly. Down and out. So notice if we were to draw these, they start at the midline. They attach to the cartilage, and their fascicles come down and out. However, remember, because they're deep, they're going to go underneath the externals. All right. So their fascicles go down and out. The externals come down and in. Notice if we look at the picture now that we know what to look at, if we look at this picture from your textbook, you see a nice job of really emphasizing these. Notice here in between the ribs, we have two sets of muscles. One is more medial, one is more lateral, one is deeper, one is superficial. One, the fascicles project down and out. The other, the fascicles project down and in. So notice we can see the external intercostals and the internal. Internal intercostals and external. External, internal, external, internal, external, internal, all the way down. But the picture I really like is this one. Notice with this one, we can't see them overlapping with each other. So we can't tell which one is superficial and we can't tell which one is deep. But notice here, we can clearly see that this one is more lateral and its fascicles are coming down and in. Notice it doesn't come anywhere near the cartilage. These are more medial, come to the cartilage of the ribs, and their fascicles are going down and out. So when you peel them apart, it's super easy to tell them apart, even if you can't tell which one is superficial and which one is deep. Being in between the ribs, their job is to help to bring the ribs up or bring the ribs down, right? So let's go back here real quickly, because we have our external intercostals, which has an E in it, an internal intercostal that has an I in it, and they both help in breathing. What did we call the two breathing processes?
Exhalation and inhalation.
Inhalation and exhalation, which happen to be E's and I's. Excellent. So that would be super convenient if the external intercostals exhaled and the internal intercostals inhaled. And is that what happens? Nope. right? Unfortunately, they do not. So we're just going to have to remember that it's opposite. Our external intercostals bring the ribs up, or more specifically, elevate the ribs. And as we know, when you elevate the ribs, this causes or leads to inhalation. So our external intercostals inhale by bringing the ribs up, whereas the internal intercostals, you stay blue. Our internal intercostals bring the ribs down. Or again, we want to be more specific, depress the ribs. And when they depress the ribs, that leads to exhalation, forcing that air out. So it'd be awesome if our externals exhaled and our internals inhaled, but it's just you're going to have to remember it does the opposite. Our internals exhale and our externals inhale. 2 more accessory breathing muscles. All right. Questions on that? Excellent. One last muscle, and this will finish us up for today. Our diaphragm is not our only diaphragm. A diaphragm is a supportive bell-shaped muscle that helps to form a boundary, and our diaphragm does that. But here, notice we have a second diaphragm. This second diaphragm is what is known as the urogenital diaphragm. There we go. Excellent. The urogenital diaphragm. Here, that urogenital diaphragm is providing the support for a female. Here we see, oops, although I got to move my name now. That urogenital, or we'll make it a tattoo. That urogenital diaphragm on a male. Their job is to help to support the abdominal pelvic viscera. Provide that support, provide that protection, keep things elevated. It plays a role in urination. It plays a role in defecation. It plays a role in childbirth. It provides that support. I know the majority of the people in this class are female, but males in high school and elementary school, when you do sports, one of the things the doctor is looking for is weaknesses in this urogenital diaphragm. And what would we call a weakness like that? We have a weakness in the wall of the muscular wall of the abdominal pelvic cavity. So fluid or mesentery, or even in some cases an organ may bulge out in that region. What do we call that?
A hernia.
A hernia, absolutely. So one of the things that boys had to do when they got their waiver signed, they had to go to the doctor, and the doctor would reach down to his goodies bits and have him turn his head and cough. Because when you cough, you increase the pressure. And he was looking for weaknesses, a sports hernia, as a result of that. This is a very important muscle that provides that structure, provides that support. And the urogenital diaphragm is primarily made by a muscle group called the levator ani muscle. Now, as you can see from the illustration here, again, both in the male and the female. Great question. Absolutely. Well, so we'll get to that in a second. So here we see the male and the female together. In both of these, we see the levator ani muscle group, but I want to go back to these pictures. Notice this one and this one. The levator ani is a muscle group primarily made up of 2 muscles, the pubococcygeus and the iliococcygeus. Now, pubococcygeus and iliococcygeus are incredibly fun to say and to spell, but are you going to be responsible for spelling those on the exam? Are pubococcygeus and iliococcygeus on your list? No. So again, this is just one of those muscles where you just need to know them as a muscle group, the levator ani. But notice that levator ani is made of the pubococcygeus and iliococcygeus in the female and in the male as well. All right, so we have that levator ani muscle providing that support of the urogenital diaphragm. Now, great question about the childbirth. I'm not sure how much it's hard to pee, but you did have hit on a key problem. One of the issues is this is supposed to be providing support and helping to sustain the organs of the abdominal pelvic cavity, of the pubic region. And the females get the honor and privilege of passing a basketball through their vaginal canal, which passes through this opening. That can cause tremendous weakness to the muscle. So I'm not sure if peeing can become necessarily challenging, but something that frequently happens for females shortly after birth is they may cough or they may sneeze or they may laugh too hard. And what happens as a result of that cough or that sneeze?
Urine incontinence.
Yeah, they can actually leak a little urine as a result of that, absolutely, because of a weakening of the wall of this levator ani muscle. This is the muscle that you normally contract to stop your urine flow, right? You're peeing and you have to stop mid-flow. You contract that muscle. That's that levator ani muscle. And so that action, that contraction, right? A female who is pregnant or even sometimes shortly after pregnancy may want to strengthen this muscle. If this muscle weakens too much, one of the things that can happen is the vaginal canal can actually descend out of the urogenital diaphragm and actually has to be surgically put back into place. So when a female is pregnant, typically she is given some exercises that they can do to strengthen this levator ani muscle. And what is that exercise that women are encouraged to do? Kegels, absolutely. Kegel exercises, right? Contract that muscle. Like I said, pretend you're stopping your urine flow. You contract that muscle, you hold it for 10 seconds, you relax, and there you go. You've just done it. Do it in sets of 10 as you do that contraction, right? And again, the nice thing about it is you can do them all the time. You can do them while you're sitting in the car at a red light. You can do them while you're standing in line at Starbucks. I just did 4 of them while I was sitting here talking to you. You can do those contractions to maintain the strength of that levator ani muscle that can help in the recovery from that pregnancy. All right. Questions on that? All right. The last thing, and I'm not going to have the time to get into all of it looking at the clock, but the last thing I want to mention, where we're going to go next is to a muscle group known as the abdominal girdle. As we talked about in the last section, and in fact, some of you had it as an essay question, our skeletal system protects a lot of the organs of our body, like our heart, like our lungs, like our liver, like some of our kidneys, things along those lines. But there's a lot of organs, the small intestine, the large intestine, the stomach, all of these things that are not protected by bones. So most of the organs of the abdominal pelvic cavity instead are protected by this abdominal girdle. This abdominal girdle is multiple layers of muscles at all sorts of different orientations to provide tension and to provide protection for the organs that are inside, right? In fact, that's how Harry Houdini died. Harry Houdini was known for having a tremendous constitution, right? He would do these big, fantastic escape artist acts, and they were incredibly elaborate setups that he would have to do. So when he was doing a show, they would have his setup, he would do one of these escapes, and then they needed time to prepare for the next one. So he would have additional acts that would come on and do their routines. But sometimes he would also come out and do things as well. He would juggle and he would do things like that. But one of the things he was most famous for is for having this great constitution. So what he would do is he would encourage anyone who wanted in the audience could come up and punch him in the stomach as hard as he wanted. And so guys would line up by the dozens to come up and punch him in the stomach. And he would engage his core, they'd punch him in the stomach, he would mock them, shame them off stage, everybody would laugh, and they would kill an hour or two while they, well, okay, not an hour or two, they'd kill, 20, 15 minutes while they set up for his next act. Well, one day he was walking out of a bar, and as he was walking out of a bar, 2 college kids who had been at the show the night before saw him, recognized him, and without saying anything, punched him rapidly 3 times in the stomach. Of course, the difference between doing it on stage when he has a chance to prepare and can engage his abdominal girdle. And when he couldn't, he wasn't aware of it. They surprised him. They end up damaging his spleen, and he died 3 days later. So that ability to protect our muscles with that abdominal girdle is something that is really, really important. And so that's what we'll talk about next. We'll talk about the 4 muscles. This is a muscle group, abdominal girdle, that we do need to know all 4 muscles. We need to be able to identify them, and we need to know their functions. All right. So there you go. But again, we obviously don't have time for that, so we will save that for next time. Excellent. So with that, that's all I have for today. Any questions before we call it a day? The spleen, I don't know the exact volume of blood in your spleen, but the spleen is a very vascularized organ. It is also a very delicate organ, so it isn't something that can easily be sewn together. And so, yeah, rupturing or damaging the spleen is ultimately what led to his death. All right. Any other questions? Okay, well, still, it's a lot. And it's very well vascularized. It's one of the most vascularized organs of the liver and the spleen. All right, excellent. With that, then, that is everything I have for today. I'm going to go ahead and stop the recording. Today's Wednesday, so have a good weekend. Study hard, study well. Start looking at that motions handout so you guys can start demonstrating some of those things. Because like I said, I think what we'll do is we'll start giving people the opportunity to demonstrate one of these activities for extra credit starting next week. So I'm not the only one here dancing around like a monkey, making fun of myself. So you guys can enjoy that as well. All right. Excellent. Any other questions? Oh, great question. So if you were doing the 5 for 5, if you've signed up and scheduled one of the 5 for 5s, you go into Confer Zoom, and in the Confer Zoom is where it should show you the meeting. So you'll click from there. I do have some times on the schedule, so both go to Confirm Zoom to schedule the appointment time, and then also that's where you'll go to make the connection link to be able to come to my office during that time. So you're not going to my main office, you're doing it to that as well. We can talk about that later. All right. Any other questions? All right, excellent. You guys have a good weekend. Study hard, and I will see you if you need to come to my office hours, and we will take care of things then. All right, take care. Bye.